Good morning, ma'am.
Good morning. Good morning. Good morning. We'll just start in a minute.
Okay, ma'am. No issue.
On behalf of the organizing committee, I, Dr. Pallavi, Assistant Professor from Department of Clinical Pharmacology, KEM, wish you all a very good morning and extend a warm welcome to the Advanced Level Training Workshop in Pharmacovigilance under the aegis of PVPI. At the outset, we welcome all chairpersons and speakers to this workshop. We are grateful to them for accepting the invitation and look forward to hearing from them. We also welcome all the delegates to the workshop. We believe that the upcoming sessions will take you through some domains in pharmacovigilance and enhance your perspective on them. These topics are not discussed widely, and we hope these will enlighten you. Before we begin, there are certain housekeeping announcements. Please keep yourselves muted at all times during the workshop. All questions from the participants should only be typed in the chat box, including those which are displayed in the YouTube comment section. The questions will be moderated by the respective chairperson at the end of each session. Now, I request Dr. Nitya Gogte ma'am, Professor and Head Department of Clinical Pharmacology, KEM, to kindly give her introductory remarks along with a brief presentation.
Thank you, Pallavi. Good morning, everybody. And at the outset, on behalf of the Department of Clinical Pharmacology, on behalf of the Pharmacovigilance Program of India, Dr. Jaiprakash Sir, Mr. Rishi Kumar, a warm welcome to all of you for this advanced level training program in pharmacovigilance. Since Pallavi has also already given an introduction to this meeting, what I thought I will do is to sort of take you through the history of Indian pharmacovigilance. All of you would be aware of the thalidomide disaster, the Kefauver-Harris amendments, and how pharmacovigilance in the developed countries in the West sort of came into being. But perhaps not all of you might know the history of Indian pharmacovigilance and how India evolved into what it is today. So I'll make a very brief presentation on the history of Indian pharmacovigilance. If I could just have my slides. Yeah. Just minimize this. Yeah. Moving forward. Let's scroll. Just move it from there. Yeah, all right. Next slide. Yeah. So what I will do is I have divided this history of Indian pharmacovigilance into 4 sections, which will be the outline of my presentation. I'll talk about what happened before we got independence, 1947. Then I'll talk about the evolution from 1947 to the mid-1970s. Then we'll take over from 1983 and go to 2009. And finally, I'll talk from 2010 to what is happening to date. Now, in 1880, this is a paper in the British Medical Journal. There was a committee called as the Glasgow Committee, which was set up by the British Medical Association, which looked at the evolution of pharmacovigilance, and they were interested in the use of chloroform. So this Glasgow Committee actually said that chloroform is injurious to the heart and more dangerous than ether. But about 8 years later in Hyderabad, in the Hyderabad Medical School, I don't know if Usha is online, 1888, there was a rebuttal by Major Laurie, who worked for this Hyderabad Medical School. He said that I have used chloroform in 40,000 patients, but I have never seen the heart seriously injured. So perhaps this was the first systematic study in India on pharmacovigilance. Next slide, please. Now, between 1947 and mid-1970s, and this is based on a literature search that I did, I could find one case series, which is PubMed indexed 1975. And this was a survey of 336 patients admitted to a general medical ward who were prospectively surveyed for adverse drug reactions. And 20% of these patients who were admitted in this medical ward were found to develop adverse reactions. Now, the true evolution of pharmacovigilance in India, in my opinion, began in the mid-80s, 1983, and then you saw it growing. This was the visionary clinical pharmacologist, Dr. Molly Thomas from Christian Medical College, Vellore, who started ADR monitoring in CMC Vellore, and have had the opportunity and the honor of meeting Madam and interacting with her on several occasions. Madam actually trained approximately 1,700 doctors in CMC Vellore. She was also trained in the Uppsala Monitoring Center. She encouraged voluntary reporting in CMC. Madam used to receive about 600 to 900 reports per year. And I remember this quarterly newsletter, which used to come out from CMC Vellore, documenting all the adverse reactions seen in the hospital. Right? Now, the mid-1980s actually saw formal action being taken by our regulator and establishment of 12 adverse drug reaction monitoring centers. And our center at KEM, it was headed by Dr. Shirsagar at that time, was one of them. So each center in India was expected to cater to a population of 50 million. And our center, well, among all the centers, of course, was linked to district hospitals. So the district's hospitals would feed into the main center. We would cater overall to a population of 50 million and then submit the adverse reactions to a database. Next slide, please. In 1997, and I happened to be in Delhi, this was the time when Professor Sten Olsen was there at the All India Institute of Medical Sciences. India became a part of the World Pharmacovigilance Program when we actually formally started reporting to the Uppsala Monitoring Center in Sweden. There were 6 centers. Mumbai continued to be one of them. And the All India Institute of Medical Sciences Pharmacology, Clinical Pharmacology departments became the national centers for pharmacovigilance in India. Now, between 1986 and 1997, apart from the efforts of individuals like Professor Molly Thomas and the regulator, there was also efforts from the Indian Council of Medical Research. Now, ICMR identified 12 centers for carrying out pharmacovigilance. There's a very nice document, a report which was compiled by ICMR. Approximately 82,000 patients were seen by these 12 centers and around 5,000 adverse reactions, adverse events documented and identified through the ICMR efforts. So there were efforts from both the Indian Council of Medical Research as well as the regulator. In 1998 saw the establishment of an independent organization that came out of Aligarh, that is the SOPI, Society for Pharmacovigilance India, who continue to have their annual programs on pharmacovigilance even today. Now, November 2004, and again, I was in Delhi at this time, saw the rechristening of pharmacovigilance, and we became the NPPI, that is the National Pharmacovigilance Program of India. And we received funding, India as a country received funding from the World Bank for 5 years to sustain this program. So it was expected that at the end of 5 years, we would become self-sufficient and self-sustaining in pharmacovigilance. The funding, of course, eventually stopped in 2009. So this NPPI avatar of pharmacovigilance had 2 main centers. KEM was the Southwest Zonal Center, and the Northeast Zonal Center was the All India Institute of Medical Sciences. But I think the true evolution and the true foundations and what we see today sort of came in the last 10 to 13 years. We have a stable system from the 14th of July, 2010. And we are now known as PVPI, or the Pharmacovigilance Program of India. This 2010 also saw the establishment of 3 panels. You have the Code Training Panel, the Quality Review Panel, and the Signal Review Panel. So formalized structures were set up in 2010. 2011, the Indian Pharmacopoeia Commission became the National Coordinating Center for Pharmacovigilance. 2012, we had the Hemovigilance Program of India. October 2013, we had a toll-free helpline for reporting adverse reactions. 2015, we saw materiovigilance. And in 2017, India became the WHO Collaborating Center for Pharmacovigilance and Public Health Program. So this sort of is the evolution, the history of pharmacovigilance, which I thought would be interesting for some of you. Next slide, please. And this will be my last slide. What I am doing today in my capacity as head of department is really carrying forward the legacy of people who have initiated this movement in the country, especially Dr. Nilima Kshirsagar, who headed the KEM Pharmacovigilance Center right from its inception, and of course, subsequently, Dr. Urmila Thatte. What I will end with is a quote which I read when I was a second-year resident in 1992. This was an editorial in the Lancet. And I think it's more true than ever before, because if you look at the COVID-19 pandemic and what we have gone through, this editorial actually ended by saying that pharmacovigilance is the price of freedom. Now, you might wonder, freedom from what? I think it gives you freedom from disease. It helps you to live a good quality of life. But freedom always comes at the cost of something. And the cost is the adverse reactions which are associated with drugs, right? So pharmacovigilance is the price of freedom and must continue throughout the lifespan of the drug. So with that, I'll end my presentation. I welcome all of you once again to this advanced level training program in pharmacovigilance. And I do hope over the next maybe 3 hours or so, you have a good, fruitful learning experience. So welcome once again, and I hand over now to Pvig.
Thank you, ma'am. As your student, it's always a delight to hear your presentations. It's indeed a privilege for me to compare this workshop. Dr. Jaiprakash Sir, Senior Principal Scientific Officer at PVPI, who has been closely associated with the department for more than 2 decades, could not attend the workshop due to his work commitments. But we have with us Shri Rishi Kumar, presently working as Senior Pharmacovigilance Associate at PVPI Indian Pharmacopoeia Commission, Ghaziabad. I request you, sir, to give us the IPC perspective through your presentation.
And welcome.
Thank you, Dr. Pallavi. And thank you, ma'am, for inviting me to this workshop of pharmacovigilance. Can I share my slide now?
Yes, please share your slides. Yeah. We've made you co-host, so you should be able to share your slides. It's visible, and the slides are visible.
Yeah, now my slides are visible to all of you.
Yeah, very clear. Please go ahead.
Thank you, ma'am. Before starting my presentation, I formally thank you, Dr. Bhakti, ma'am, Dr. Pallavi, and the whole team of Clinical Pharmacology, Department of Clinical Pharmacology, KEM Mumbai, for inviting me to this workshop. And on behalf of Dr. Jaiprakash, I'm going to present this Pharmacovigilance Program of India perspective to all of you. So in my presentation, I will focus on the functions of Indian Pharmacopoeia Commissions. What is pharmacovigilance? As you all very well know, we will not go in detail of this particular point. What is the need of pharmacovigilance? That what is next? The progress of pharmacovigilance program of India. What are the minimum requirements to report adverse drug reactions? How the data is being flowed in the pharmacovigilance program of India? ADR reporting status of India as on date. What are the various channels of ADR reporting that Pharmacovigilance Program of India is using? What is our status of India at global level while reporting adverse drug reactions to the VCPs? We will also touch upon what are the outcomes of this database till date that we are having. And apart from that, the various activities, various training programs that we have done till date on the from the Pharmacovigilance Program of India point of view. Basically, Indian Pharmacopoeia Commission is an autonomous organization of Ministry of Health and Family Welfare, which have basically 4 major functions. First one is publication and releasing of Indian Pharmacopoeia, which is a book of standards and which is legally applicable in case in court of law in case of any legislation due to use of drugs. Second most important function is National Pharmacology of India. Indian Pharmacopoeia Commissions is publishing this official document for the pharmacists or healthcare professionals for rational use of drugs. Third main function is the releasing of reference substances, which is used for analytical purpose in various pharmaceutical laboratories. And the fourth main function is Pharmacovigilance Program of India that already we are as a national coordination center since 2011, and we are progressing well, as Dr. Bhakti ma'am also gives some light on this point. Apart from these 4 functions, Indian Pharmacopoeia Commission is also responsible for trainings of drug regulators, and also we are responsible for testing of national drugs, new drug substances. It is an additional activities given by DCGI to this office. And recently, you all know very well aware about the cough syrups tragedies throughout the world being reported due to the ethylene glycol toxicity. So Indian Pharmacopoeia Commission's laboratory has been chosen by the Ministry of Health and Family Welfare to test all those cough syrups, specifically paracetamol cough syrup, syrup and other cough syrups need to be tested for DEZ at various laboratory in India, and Indian Pharmacopoeia Commission's laboratory is one of them. So these are the few functions of Indian Pharmacopoeia Commission that we are doing. If we talk about pharmacovigilance, it can be summarized in 4 major points, that is detection, assessment, understanding and preventions of adverse effects or any other medicines related problem, because this is basically the science and activities which relate to the detection, assessment, understanding and prevention of adverse drug reactions or adverse events. Why we need pharmacovigilance? To protect patients from unnecessary harm and increasing healthcare costs from medicines. As we all know, adverse drug reactions are a huge burden on patients and on the country also, any country. Insufficient evidence of safety of drug from clinical trials, because we all know that during the clinical trial, it's not possible to collect all the adverse events. Some of the adverse events seen after the clinical trials during the phase 4 studies, that is post-marketing surveillance. For signal detection, risk minimization measures, we need pharmacovigilance. Earlier detection of new ADRs on patient subgroups of exceptional sensitivity like pediatrics, geriatrics, and pregnant women. Promoting rational use of medicines and medication adherence. To support regulatory agency in decision-making process for safe use of medicines. Because before 2010, whenever we need to take any decision on the regulatory sides of the medicines, we look towards the data of West. But today, we can say that we have handsome amount of data based on which we can analyze any drug in India based on the India specific populations if we require to take any regulatory decisions on our medicines. It subsequently it boosts the public confidence for safe and proper use of medicines. The journey of pharmacovigilance, it already has been covered by Dr. Nitya Bhakte very nicely. So I will not go in the detail of this particular slides. That pharmacovigilance program from India, as ma'am has already told, that that program was launched in 2010 by Ministry of Health and Family Welfare at KEMH. Subsequently, it was transferred to Indian Pharmacopoeia Commission on 15th April 2011. After that, in 2015, the Material Program of India was launched. And in 2012, Hemovigilance Program of India came into the picture. And in 2017, WHO Collaborating Centers was established at IPC for public health programs and regulatory services. So starting from 22 centers in 2011, at present we are having 760 area bonding centers all over India. And we cover almost all states and all union territories except one, that is Lakshadweep. We are in the process, we are reaching out to the authorities to establish one ADR mounting center, at least one ADR mounting center in that also. And all the efforts are on the process. And apart from that, we are having, currently we have received in this till this month 80 letter of enrollment form from various hospitals all across India. So probably in this month our strength will be 840 in the coming month. So apart from all these efforts, Ministry of Health and Family Welfare has asked us to submit a proposal for enrolling all central government hospitals, including railway hospitals and army hospitals and all CGHS dispensaries. So we have submitted the proposal, and in the coming months, or coming years, our network of pharmacovigilance will be a strongest one in the world. I could say it will be strongest one because all the hospitals will be covered. It already has been supported by National Medical Commission by issuing a public order to all accredited or all approved medical institutions in India to become ADR mounting center. So all of them are already ADR mounting center. Few are remaining, near about 40. If I say, we will receive letter of enrollment subsequent to this letter issued by National Medical Commissions. Apart from that, I would like to inform all of you that IPC is having a memorandum of understanding with National Accreditation this NADH, National Accreditation Board for Healthcare Providers, hospitals and healthcare providers. That memorandum of understanding has been renewed recently in the past month, and now they have assured us that all the NABH accredited hospitals will be our ADR counting center, and they will start reporting. They will start doing inspection on these hospitals whether they are reporting ADRs to PVPI or not. So that will be one criteria in their inspection list. So this is our this is the expansion of PVPI in all states and union territories all across India. This is the representation of our ADR mounting center at various states. So why do reports? Because need of pharmacovigilance I already explained, and one person can make a difference if that particular adverse drug reactions is a new one. So it may lead to change in package insert like that. It may lead to issue a drug safety alert, or it may be a signal after analysis. So who can report? All consumers, patients, physicians, pharmacists, all allied healthcare professionals, pharmaceutical industries can report ADRs to PVPI. What to report? All type of suspected adverse reaction, known or unknown, serious or non-serious, frequent or rare, reactions from all type of pharmaceutical products like allopathic product, Ayurvedic product, vaccines, medical devices, extra, can be reported to PVPI. How and whom to report? As I told you, we are having various ADR mounting center. Presently, it is 760. That ADR can be reported through these ADR mounting center. Apart from that, we are having other modes of reporting like mobile app application, ADR PVPI. It is freely available on the Google Play Store. Anybody can download in an Android mobile phone and report adverse reactions. Other than this, all healthcare professional or consumers can report to helpline, which is toll free 1800-180-3024. It is available from Monday to Friday, 9 to 5:30. Apart from that, any stakeholder can report through ADR reporting form. We are having two type of form. One is for consumer, which is very simple form, easy to understand and available in various vernacular language, available on our website, and we circulate it through our ADR monitoring centers also. And second form is red color form, which is for healthcare professionals, can be filled by healthcare professionals and can be sent directly to NCC or to the direct nearby ADR monitoring center. So minimum requirement for reporting of areas, basically it is having four criteria: an identifiable patient, an identifiable reporter, an event, and a medicinal product. That should be there in any valid ADR report. These are two forms. Blue color form is for consumer and red color form is for healthcare professionals. And this blue color form is available in 10 regional languages. This is the data flow in pharmacovigilance program of India. First of all, data is being getting through from ADR monitoring centers, which is currently in 716 number. It is being reported to NCC PPIs through a software known as VisiFlow, which is provided by WHO Collaborating Center, Upasana Monitoring Center. Further, the data is being analyzed at NCC PPI, and if we found any new adverse drug reactions or any suspected adverse drug reaction, any SHUSA, suspected serious, unexpected serious adverse drug reaction, that we are going to put in to the signal review panel, and signal review panel further analyze the data, and based on their analysis, they further recommend it to CDSU for regulatory action. So how to become an ADR monitoring center? NCC PPI seeks letter of intent from dean, head of the institution, hospitals, for establishment of on AMC first. Second, NCC PPI examine suitability of the proposed center on the basis of the performance. Third, approval from the competent authority. Fourth, communication to the newly inducted AMC. So basically, these are the four steps that we do to enroll any ADR monitoring center under the flap of PVPI. Apart from that, we are having collaboration with various public health programs. As I told you that we are the WHO collaborating centers for public health programs and regulatory services. Basically, we are having collaboration with four national health programs or public health program. One is NTEP, National Tuberculosis Elimination Program. In 2014, there were 21 centers, and with the pass of time, we are receiving letter of enrollment from other TB centers also. They are reporting to us. Further, we are having collaboration with National AIDS Control Program centers. Presently, 20 centers are reporting to us, and through our ADR monitoring centers, because these ART centers and NTB or TB centers are placed in the various medical colleges, and through our ADR monitoring center, we are receiving report from almost all TB centers and these ART centers. We are having a collaboration with National Vector-Borne Disease Control Program, currently 58 centers. Basic aim was to collect the reports of drugs used for the treatment of Kala-Azar, and we are receiving these reports from 58 centers at NCC. Next is Universal Immunization Program. Through UIP, through network of UIP, it is one of the largest program of India, through which we are receiving adverse drug reaction related to vaccine in our database. And it has been already covered. This is ADR reporting status. At present, till 16 June 2023, we have collected 7,12,047 ICSR in our database. And in 2023, it already have been crossed 50,000, and we hopefully will cross the mark of 1,17,000 for of the last year ICSR submission to the WHO. These are the data of helpline that we have received since the inception of helpline, and it is also increasing year by year. ADR reporting via mobile app is also increasing. This is the data of ADR reporting through mobile app. These are the data of training organized by Pharmacovigilance Program of India. And in last 2 years, now 2-3 years, the number of training programs has been increased. This all because of efforts by our core training panel. They have mandated our pharmacovigilance associate to conduct at least one training program in a month. And apart from that, our regional training centers are also doing exemplary job in part of training programs, and this is the training program we all are attending is one of the example in that part. And these are the outcome of Pharmacovigilance Program of India. Till date, we have issued 153 drug safety alerts. We have issued 56 PIL changes and 12 signals. So overall 68 recommendations have been raised to CDSO, and almost all have been approved by CDSO expert committee or subject expert committee to be implemented at the ground level by the state drug regulators. Now these are the signals that PVI has issued 12 signals, and the last signals was by the paracetamol, but it was fixed drug options by paracetamol. These are the drug safety alerts in year in last 1.5 years, you can say. In the last drug safety alert that was issued on 31st May 2023 was electrocardiogram QT prolonged or QT prolongation by the use of ceftriaxone. It's a drug safety alerts. We encouraged our healthcare professional to look on this particular event, and if they came across any such events, kindly report to NCCP by various means of reporting. Next is levosulfride. This event name is restless leg syndrome. It was issued in the last month, 31st May 2023, and same is expected by our healthcare professional to keep watch on this particular drug to this particular event. These are few outcomes have been outcomes of PPI has been published in the WHO pharmaceutical newsletters in the last year. This is the quality of ICSR submitted to WHO, and this blue line indicates that the quality of ICSRs from NCC PPI is quite better as compared to the green line, which is the completeness score of other countries which are reporting to UMC. Our quality of ICSR is good because of our ADR monitoring center, because of our pharmacovigilance associates, because of our AMC coordinators. They are looking after the data that we are collecting at AMC and submitting to NCC, subsequently to UMC. These are all team efforts that our completeness score is around 0.77 out of 1. These are some PPI resource materials. Is every quarterly PPI used to issue newsletters and annual report and once in a year. Till date, our team has conducted 5,993 training and sensitization program, in which more than 3 lakh healthcare professionals and stakeholders have been trained for PV activities. Globally, if we see the data of India, it is 2% only, and which indicates that there is a large scope of improvement in terms of ADR reporting, because India is the largest populated country in the world, and our contribution is merely 2%, since whereas our contribution to the population is significantly high. So we must be in the top 2-3 positions, and our efforts are towards this direction. Soon we will be, and we are going to be on 8th position, and because our ADR reporting is increasing day by day. This is the letter issued by National Medical Commission to all medical colleges to become ADR monitoring center. Already explained. One major activity that we are doing is skill development programs. It was started in the year 2017, and frequency of this training program is 4 times in a year. It is a request to all participants. Please convey this message to all your friends or colleagues in the medical fraternity to attend this program. This is at present being conducted virtually by the National Coordination Center, and it is a very good program for the beginners who want to make their career in pharmacovigilance. So these are the number of participants when pre-COVID and post-COVID comparison have been done. In pre-COVID, because program was the physical one, and total number of participants was 465. But when the program was started virtually, the number of participants has increased significantly. It is number till date is 1455, and the next training and next skill development program is going to be happen from 7th August to 11th August at IPC. The last date of application is 2nd August 2023. So I request everybody to convey your friends regarding this skill development program so they can be benefited by these programs. So with this, I end my presentation, and thank you all. If any questions from the participants, I'm happy to answer.
Thank you very much, sir, for providing the comprehensive, clear, and precise perspectives of IPC.
Thank you.
We are currently having 100+ participants on YouTube with us. So any question? No. So no questions for this session. So now we will begin with our scientific sessions. Going towards the first session of the day, I invite Dr. Usha Rani ma'am, Professor and Head of Clinical Pharmacology, Nizam's Institute of Medical Science, Hyderabad, as the chair of the session. She is a well-known name in clinical pharmacology and a close friend of the department. Ma'am currently also holds the position of Executive Committee Members ISCR, Member Secretary, NIMS Ethics Committee for Stem Cell Research, Certified NABH Assessor for Ethics Committee, Clinical Sites and Investigators, Member Deputy Task Force for Translational Research, and Member IMA Standing Committee for Pharmacovigilance. In her illustrious career, ma'am has been the member of various national and international societies, regulatory committees, and editorial boards of various national and international journals with 150+ research articles to her credit. It's a matter of pride to have ma'am with us today. I request ma'am to kindly introduce the speaker and take the proceeding forward. We also request you to monitor the chat box for discussion at the end of the session. Comments received on YouTube will also typed in the chat box for you. So over to you, ma'am.
Yeah, thanks a lot, Nitya and team, for giving me this opportunity. And it's a pleasure welcoming Dr. Prasad Kulkarni to speak to us on vaccine safety and overview. Dr. Prasad Kulkarni is Executive Director at Serum Institute of India Limited, and is a clinical pharmacologist with several years of experience in new drug development, pharmacovigilance, clinical research, regulatory practices, and he has been instrumental in successful clinical development of several new vaccines and recombinant products, proteins at the Serum Institute. He has more than 70 publications to his credit in various national and international journals, and naming a few of them like the NEJM, Lancet Infectious Disease, Clinical Infectious Disease, Vaccine, and so on. And he's been a faculty at various national and international conferences. So welcome to you, Dr. Kulkarni, and over to you. And I request you to give your presentation on vaccine safety and overview. Thank you.
Thank you very much, ma'am, for the kind introduction, and good morning, everybody. I'll be taking you through this vast topic, and it's not actually possible to cover in a 20-minute session, but I will try to do some justice. Can you see my screen?
Yes, Dr. Kulkarni,
Yeah,
please.
yeah, you're visible. Yeah.
Yes. Okay. One moment. I'm trying to get this. Yeah. So vaccines, as you know, that it's a preparation that is used to stimulate a body's immune response against diseases. And once vaccine is given, it produces something called as immunity, which is a protection from infectious disease. And you can get this immunity either by getting a vaccine or by getting natural infections, right? So but why wait for the natural infection? Because natural infection obviously can cause severe devastating effects, right? Including death, for example, like we have seen in the case of COVID. So that's why we need to prevent those infections so that we get immunity, artificial immunity, and get us protection from the infections. So why they are special? Because they promote health, obviously. They prevent almost 5 million deaths every year, and they have huge reach. They can go to populations, they can go to the entire communities, countries, etc. And smallpox eradication in 1979 is a classic example of how immunization can really completely eliminate a disease from the face of the earth. They have very rapid impact, and within few years you can actually eliminate one infection from at least one part of the country. So for example, polio, we have seen that it's almost on the verge of elimination through sustained immunization, through routine, as well as through pulse polio. You must be seeing that. And what is the effect of this? They save lives, obviously. They save a lot of money, a lot of sufferings, right? You don't see any crippled child anymore, right? When we were children, we used to see a lot of crippled children taking walking sticks because they were affected with polio. Measles similarly used to kill a lot of children. So this is just to show you the impact of the vaccines, starting with smallpox, diphtheria, measles, mumps, etc. So these were the 20th century annual morbidity. This you can say like a pre-vaccine era, when those many cases used to happen, and this is only for USA, mind you. So even USA used to have a lot of infectious diseases before the vaccines got introduced. And now, this was in 2021, almost all of these diseases have disappeared from USA. So the decrease in terms of percentages has been phenomenal. And this has been possible, obviously, because of vaccines. So this is what CDC says, that it prevents so many illnesses, millions, so many deaths are prevented, and trillions of dollars are saved every year, which otherwise would have been spent in treating those cases. COVID vaccination in India, for example, has been one of the greatest public health success stories. And the latest data that came from Stanford University showed that it actually saved India as a country 1.29 lakh crore, right? That's a huge amount that can run the country's budget. And also, obviously, it saved millions of lives, right? Almost 4 million lives have been saved because of vaccination. So that's why vaccines play a big role. And also because of the same thing, vaccine safety becomes so important, right? Because unless vaccines are safe, people will not have confidence in them. People will not take the vaccines. If they don't take the vaccine, the infections will happen and will cause a lot of havoc in the community. And therefore, we need to be vigilant. We need to be really careful, especially because vaccines are also prone to a lot of rumors. We know that we have seen that, especially during the pandemic, the WhatsApp University had circulated so many, news about different vaccines, and so many people were actually not ready to take the vaccine. Many people did not take the vaccine, thinking that they will cause more harm, and some of them actually got the infection and also died. So that's the reason why you need to be really vigilant, and this kind of false claims of EFS can really create havoc. One prime example was in 1999, when 1998, when this Lancet paper was published by a pediatric gastroenterologist called Andrew Wakefield from UK. And in this paper, he claimed that in his study, MMR vaccine led to autism in the children. right? So that was a very big claim. And that time, the autism cases were getting diagnosed more and more in the Western world, especially in USA and UK. And this created havoc. And naturally, people stopped taking MMR vaccine. And that really had a big problem. It resulted into measles outbreaks in all of the Europe and also in US. And many children died because of that, right? So such false rumor. And obviously later on, it was found out that the paper was absolute fraud, and he had made these false claims. The paper was retracted by Lancet, and his license to practice was cancelled, and he became a completely, a persona non grata in the scientific world. So, but by that time, he had already created damage, and some children had died, and there are still some survivors of measles who got encephalitis or SSPE, who are having lifelong problem. Who was responsible? This guy was responsible. So any such false claim can really create a lot of problems. Like in Nigeria, for example, they many of them boycotted the pulse polio programs. Why? Because they said that the oral polio vaccine can cause infertility in their babies. And this rumor was circulating for a long time. And because of that, polio continued to happen in Nigeria, also continues to happen in Afghanistan and Pakistan because of the same rumor. So you can imagine the consequences that vaccine rumors can have. And therefore, we have in place pharmacovigilance, as Sir already told you very elaborately, that how our pharmacovigilance system is in place. The country's system is working so well. And as a part of that, you have to keep safety surveillance in place and also safety communications. So before getting into that, we need to understand something called as AEFI, adverse event following immunization, which is any untoward medical occurrence that happens after a vaccine, but need not be caused by the vaccine. That's very important. So anything that happens is not caused by the vaccine. That we must remember. Today I took the vaccine, tomorrow I got some brain infection, does not mean it was caused by the vaccine. So this is something called as temporal association. But what we need to look for is the causal association. Is it really caused by the vaccine? That's what is more important. And that's why any science or activities relating to the detection or understanding communication of AEFI or any other immunization issues, so that we can prevent untoward effects of the vaccine, is called as the pharmacovigilance. And there have been, since ages, there have been something called as anti-vaxxers, right? These are anti-vaccine groups, very strong groups, very well-funded groups, and they are the ones who create panic among the public. So you see the cartoon here, vaccinations can be dangerous to our health, and this kind of group, they continue. So why do you need pharmacovigilance for vaccines? Because there are some basic differences between vaccines and drugs. The first important is healthy people get vaccines, including the smallest infants and newborns, whereas the drugs are given to sick people. That's a very basic difference. Because you want to prevent a disease, you give vaccines, whereas drugs are given to treat a disease. And their vaccines are given to a large number of people at one time, whereas drugs are given only in hospitals or only in those who go to clinics, so small number of people. And vaccines are given at certain time points, like during the first year of life, etc., where there are other things that are also common, like other infections, for example, diarrheas, pneumonias, they are common, or some congenital malformation or something like that. Whereas drugs are given only when you fall sick, right? There is no particular time as. So because of all these factors, the tolerance for adverse events is very low. The acceptance for side effects with vaccines is very low. You don't want anything to happen to a healthy baby, right? He was healthy, and now you get the vaccine and it caused harm to the baby. That's not acceptable. Which is what the psychology is, which is correct, which is perfectly correct. You don't want to make any healthy child, any healthy person sick, right? Whereas in case of drugs, the person is already sick. Let's say as somebody is having cancer, he gets an anti-cancer drugs. Those drugs are having severe toxicity, but the patient will tolerate that. Why? Because the underlying disease is so severe that he is ready to accept anything any kind of toxic effects because he thinks that it will cure my cancer. So that's not the case with vaccines, right? And so that's a low tolerance, and many vaccines are given, as you know, during the childhood. So safety of the vaccines is monitored throughout the life cycle, starting from the quality tests at the industry level, then non-clinical animal studies, clinical trials, and there is elaborate batch-to-batch testing that happens during each vaccine batch that is produced. Each vaccine batch in our country, for example, is not only released by the company, but also has to be released by the Central Drugs Laboratory, CDL Kasoli, which is a government lab. It's a national control lab, right? So that's an additional safeguard that is kept in case of vaccines, unlike the drugs. Drugs, every batch doesn't get tested by any regulatory laboratory. So there is elaborate system in place in terms of vaccine safety surveillance. And during the pre-licensure period, for example, before getting the vaccine, the vaccine gets tested in phase 1, phase 2, phase 3 trials through different number of individuals, and then all that data is seen by the drug controller, and only when they are convinced that the vaccine is safe, then they give licensure. So this is about the pre-licensure. But even after the post-licensure, we know that you have to have pharmacovigilance in place, and we know the reasons already. We have heard already that. But in case of adverse events following immunization, we need to keep something in mind called as causality assessment. And WHO has made a classification of any AFI. They have made 5-6 categories based on the causal association. So they can be the vaccine really caused by the vaccine. So vaccine product related reaction. They can be because of the quality defect in the vaccine. So something is wrong with the vaccine. Then there can be immunization error. So when the nurse is giving the injection, whether she has made any mistake. It can be because of anxiety, right? Because many people are anxious to get an injection, so anxiety can cause. Then there can be coincidental events which have nothing to do, and then there can be some events where you don't know, they are indeterminate. So vaccine product-related reactions are because of the inherent properties of the vaccine, right? They can be because, let's say, measles vaccine, if you give, it is going to cause fever and some rash in some children. Right, that is happened because it's a live vaccine. That's an inherent property of the vaccine. Can be because of the route of the vaccine or the site of the vaccine. For example, if you give DPT vaccine, it's going to cause some local redness and swelling and sometimes nodule in some children because it is aluminum as an adjuvant. So that is going to cause some local reaction. So that's bound to happen. Sometimes immune-mediated reactions, for example, BCG vaccine, which is a live attenuated Mycobacterium bovis vaccine, it can cause lymph node swelling because of the immune response. Sometimes we saw that because the replication of the vaccine virus, like in case of measles, it can mimic the original infection, obviously a mild variety. So how do you determine this, whether it is really caused by the vaccine? So you have to look for whether there is a temporal association. Obviously, it has to happen after vaccination, whether it is biologically plausible, right? There has to be some biological mechanism which can explain the reaction. Whether it is consistent, you have seen it in different population across different countries, different time zones, different years. Whether the strength is there is a strong association, whether there is dose response, whether it gets reversed, and all that. So these are the basic factors based on which you can determine whether a vaccine has caused that reaction. And we know for most of the vaccines, we already know what reactions are caused by them. For example, local reactions are caused by almost all the vaccines like BCG, hepatitis B, MMR, polio, pneumococcal, tetanus, etc. Many of them also can cause fever, right, at different frequencies. So the frequencies are also known. If it goes beyond that, then you could be worried, right? For example, let's say hepatitis B causes 1 to 6%, and then let's say in a center you get half of the children getting fever, you will be worried. You will try to investigate that, right? And also some of them can cause systemic reactions like irritability, malaise, etc., etc. So this is already well defined, right? Because of the data that has been collected. We also know that the side effects that are caused by the vaccines are much, much less as compared to the infection, natural infection. So this is a comparison between infection, measles vaccine. So infection can cause otitis, pneumonia, diarrhea, encephalomyelitis, SSV we talked about, subacute sclerosing panencephalitis, anaphylaxis, of course not, but thrombocytopenia and some antis as well. So these are the complications of measles. Whereas the vaccine will not cause almost all of them. So very rarely it can cause anaphylaxis. Extremely rarely, now this is also gone. It doesn't cause encephalitis, right? Thrombocytopenia it may cause. So all of this that can happen with infection, most of it will not happen with the vaccine. So this is the risk-benefit analysis, correct? Benefits are far, far more than the risk. Serious reactions can happen, yes, definitely, but they're extremely rare, extremely rare. For example, disseminated BCG infection can happen absolutely extremely rarely. Vaccine-associated paralytic polio with OPV, or prolonged crying or seizure with DPT, measles can cause seizure, anaphylaxis, which is the life-threatening one, right? That can happen in 1 lakh. Intussusception with flu rotavirus vaccine can happen extremely rarely. So the chances of those reactions are very rare. Moreover, they can be easily treated and the deaths can be avoided. There are no deaths possible. Whereas the infection can actually cause deaths, right? Quality related reactions, as we talked about, can be because of the defective quality. That can happen extremely rarely. Generally, nowadays it does not happen. Long back in 1955, a killed polio vaccine actually contained live polio virus vaccine, live polio virus, and that caused polio because of the injection. That was in USA almost 70 years back, right? Since then, there has been no real quality defect that has been seen with the vaccine. So that is almost not possible. Immunization errors continue to happen, right? And what are these? For example, you give it by wrong route, oral polio vaccine being given by injection, right? Nasal vaccine given orally. Anything is possible. And when you give vaccines to millions of babies, some errors will happen. Or using the wrong diluent. For example, in Syria, during the war in 2014, they use measles vaccine, and with measles vaccine, it comes as a powder which has to be diluted with the diluent, which is sterile water for injection. Instead of using that diluent, those people use a muscle relaxant, which is used as a pre-anesthetic medication. And they diluted with that and injected babies, and 15 babies died because of that. Because of the muscle relaxant, their respiratory muscles went into paralysis and they died because of respiratory. So these kind of errors can happen. Measles vaccine being reconstituted overnight and then used next day. By that time, Staphylococcus aureus grows in the vaccine. You inject that, the toxin comes in the baby's body, and babies die because of something called as toxic shock. So these kind of errors can happen. Anxiety is also very common, especially in older children, in the adolescent age group, especially in girls, that can happen, and they can feel giddy. Some of them fall because of vasovagal syncope, etc. And many of the reactions that were reported to government after COVID vaccination, many of them were because of anxiety, right? We know that people started connecting anything and everything with the COVID vaccine. And then there are coincidental events which happen whether you receive vaccine or not. And many of the events are also coincidental. For example, sudden infant death syndrome is a known event that happened during infancy, but because they happen after vaccination, they have been connected with the vaccine, especially with DPT. But they are coincidental. They don't happen because of the vaccine. And sometimes, of course, you don't know what is exactly the cause of the vaccine. So they are indeterminate, right? And so these are the categories of various vaccine-related EFIs. And that's why in India, we have a huge safety surveillance. India's universal immunization program is the largest in the world, and millions of babies are targeted every year. These are the various vaccines that are given in our national immunization programs. You can imagine when 27 million babies are born every year, there are millions of vaccines that are being given. We already saw the milestone of AEFI surveillance system in India, and we have a AEFI surveillance organization structure as well, very elaborate, very nicely working. So it starts at the basic level, an AEFI, then goes to district, state, and then finally the Ministry of Health. And there is an elaborate investigation for any AEFI that happens. Globally, also, there are multiple bodies that are working towards AEFI surveillance, like WHO, then SIAMS in USA, you have VAERS, Canada, Australia. Every country has got a huge mechanism in place. And also the safety surveillance is very important during the mass immunization campaigns like that happened with either pulse polio or our COVID vaccination program, which was also an immunization campaigns. And there are specific challenges because when you give so many doses in a very short span of time, naturally you're going to get a lot of reports, right? And when there is enhanced surveillance in place, you're going to get more reports. Not all are caused by the vaccine. That's what you need to keep in mind. Pregnancy is a special consideration. There are vaccines like flu and TT, which are recommended during pregnancy so that you can prevent the complications in the babies. All live vaccines are contraindicated, that we need to make sure, and other vaccines, the physician had to make sure that whether the risks are more or the benefits are more. So to avoid all these problems, you need to have safety communications in place. We need to tell public that no vaccine is without any risk. Obviously, most of the risks are absolutely minor and they need to be communicated. But a rumor can really cause severe problems, and that's why you need to stop. Sometimes you need to stop the vaccination, you need to stop the immunization program, and that creates a problem. So proactive communication is very, very important. What should you tell in the communication? The benefit risk balance. That's very important. That how the benefits are much, much more than the risks, right? And why you are giving this communication, how you want to deal with this safety concern, whether this safety concern, whatever is there, whether it is real or it is just a hypothetical one, and all that along with scientific evidence, right? Everything has to be evidence-based. So, and why is this necessary? Because vaccine hesitancy has now become the top 10 threats to global health, right? WHO has clearly said that. And what is this hesitancy? That is delay in acceptance or refusal of vaccines despite the availability of vaccination service. And that vaccine hesitancy can create havoc. These are the pictures from USA during the COVID pandemic. Wake up, America. We have the sickest children in all the developed world. Vaccines trading mild illnesses for chronic diseases, right? COVID, 99% recovery with no vaccine. Why? Gates of hell, Bill and Melinda. So Bill Gates Foundation was a big target during this entire hesitancy thing. Freedom is not negotiable. Government is lying to you. Say no to the COVID vaccine, save the children. There have been a number of agitations and demonstrations in USA. A number of people did not take the vaccine. And what was the end result? USA is the only country which had 1 million COVID deaths. 1 million COVID deaths. No other country, even India, didn't have so many deaths. Why? Because of the vaccine hesitancy. People just did not take the vaccine. So that's the price that you pay if you are ignorant, if you don't pay heed to the scientific evidence. What are the causes of the vaccine hesitancy? Multiple, basically rumors, media, safety related, efficacy related, perception. We saw already, right? COVID gets cured on its own. You don't need a vaccine and all that. It also depends on the education level of the people, their income level. Poor people, low educated people are more prone to this kind of perceptions. So we need to have a multi-sectoral approach. You need to involve the healthcare professionals, media, social media, policymakers, governments. Everybody needs to come together so as to dispel any of those myths. And you continue to give recommendations through basic communication that vaccines are safe and there are nothing to be concerned about it, and they save lives, right? So these are some of the social media campaigns that are run by UNICEF, for example, right? Just a few, and why parents don't vaccinate and outbreaks happen, or don't let cervical cancer stop you, get vaccinated. Vaccinate children against measles, vaccines save lives. This is from WHO, WHO again, and PAHO. for polio vaccination. This is another communication from AstraZeneca. If you remember, with Covishield, there was a reaction called as thrombosis with thrombocytopenia syndrome, a clotting problem that killed few people. But this was a communication that dying from TTS is 1 in 1 million, whereas death from TTS is 1 in 1 million. How small is 1 in a million? So the risk 1 in a 1 million is how rare? It's extremely rare. So if you can see, this is like one single hair out of total 10 people's hairs, right? 1% out of 10 sold out MCG stadiums. 8 centimeter a long way from Brisbane to Gold Coast, which is 80 kilometers. So that's how the small risk is. Or risk of death dying from a general anesthetic is 18 in a million. The risk of death from accidental poisoning is 55 in a million. The risk of dying because of lightning is 2 in a million. Risk of dying due to rock climbing is 3 in a million. So there are other things which are actually more risk prone, and TTS was such a rare. So that's why communication is so important. Coming to the end, vaccination is one of the top public health achievements of human history. They are safe and effective. Their safety is continuously monitored throughout their life cycle. They give confidence to the healthcare workers, regulators, and to the public. And strong AFI surveillance and communications are essential for successful immunization programs and ultimately for prevention of various infections. So thank you very much. And I'm open to some questions.
Thank you, Dr. Kulkarni. You've very clearly elaborated on the vaccine safety. And we all agree that AEFI causality assessment is something which is extremely important, because many a times, the causality is given wrong, and that causes a lot of problems. And then communication, yes, very true. There should be proper communication, should be an open-ended. And your experience, anything on the study designs, like what are the problems you faced, especially with vaccines?
Well,
a couple of points.
Yeah, I mean, I can give the recent example of Covishield, for example, that during the trial that we were doing, there was a case of multiple sclerosis that happened in the UK trial, the study that was being done by Oxford University, and there was an investigation and the trial was paused just as a precautionary measure. And the news obviously flashed across, and then immediately there was huge concern in India that this is a problem, could be because with Covishield, and we also had to stop the trial. And finally, when the UK regulator investigated the case, it was found that the case had nothing to do with the vaccine. The trial resumed, and so did our trial. But we lost a few days in that process, right? So these kind of things can happen, and we need to tackle them at that time, So there are, of course, several examples, but this is the most recent one.
Yeah, I know it has to be tackled case by case basis. And since we are running short of time, there are a couple of questions in the chat box. It says that during the pandemic, vaccine development was extremely fast. So how do you establish a robust pharmacovigilance program in that scenario?
Right. This has been a very, very common question asked. So we need to keep in mind that vaccine that were developed for COVID, the platforms were already ready, the vaccine platform. So this is what takes a lot of time. And because the platforms were ready, so they could be easily translated for making the COVID SARS-CoV-2 vaccine. So that didn't take a lot of time. The regulatory approvals came in very fast in all the countries because this was a pandemic. It was a global emergency. And the trials did not take a lot of time because the efficacy could be achieved very quickly. See, the efficacy trials is the one that takes a lot of time, 2, 3, 4 years, because the infections happen at a very low attack rate. You don't get to see the cases very quickly. But that was not the case with COVID. The COVID cases were happening so quickly that you got the required number of cases very fast. And that's how you could reach to the efficacy conclusions fast, and you could get the required data. And therefore, because all that happened within a year's time, you did not get the full license. You got emergency use approval. It was an EUA, right? So it was not a full approval. And therefore, during the entire campaign that happened, there was a very big surveillance in place just to make sure that, in fact, this kind of surveillance, AFI surveillance, has not been there for any other product in the world, never in the history. And I can very confidently say that the amount of safety data that has been collected for COVID vaccines is not available for any other vaccine. Within this short span of time, if you go to the literature, you will find thousands and thousands of papers on the safety of this vaccine, huge safety surveillance. So all that risk, probably the hypothetical risk that we were taking by giving a fast approval, was taken care by keeping a very elaborate surveillance in place. So I don't think we really were required to worry about that, and we have been proven right. The vaccines have been proven very safe, except for a few extremely rare events, and they have saved millions of lives.
Nitya, do we have time for questions or?
Usha, there's one more short
Yeah.
question. Then we can.
Okay. Yeah. So the second question is, in your opinion, from your experience, what is the proportion of AEF5 which are due to errors and quality issues from your database?
So quality issues, I already said that they are not seen any longer, because the quality systems that are at the manufacturer's ends are very, very stringent. And moreover, as I said, the National Control Laboratory in Kasoli, they also release the batches of the vaccine. So there is a double layer quality mechanism in place. So you no longer see those kind of cases, except for the 1955 case of polio happening. That is that. And in errors are also very rare. Definitely they are very rare, but they can happen. And probably they will keep on happening, unfortunately. And that's why you need to keep on training your manpower and your healthcare workers. And that government does that fantastically. Our immunization system is extremely well trained, and that's why errors especially in India, are extremely rare.
Yeah, true. Yeah, I think thanks, Dr. Kulkarni, and thanks, Nitya and team, for giving me this opportunity. Thank you.
Thank you, madam. Thank you, Dr. Nitya.
Thank you, sir, and thank you, ma'am. We hope the first session has been a learning for all the attendees. We move ahead to the second session for the day. I invite Dr. Urmila Thatte, ma'am, as the chair of the session. Dr. Thatte, ma'am, has previously held the position of head of Department of Clinical Pharmacology, KEM, and is presently emeritus professor of Department of Clinical Pharmacology, KEM, Mumbai. Ma'am has served on ethics committees of KEM Hospital as well as independent ethics committees in Mumbai and Bombay hospitals, and is secretary of FERCI, that is Forum of Ethics Review Committees in India. She has over 260 publications in national and international journals, including 10 books to her credit. She was awarded as top 2% scientist in the world for 2 years consecutively by Stanford University. It's a matter of pride to have Madam with us today. I request Ma'am to kindly introduce the speaker and take the session forward. We also request you to monitor the chat box for the discussion at the end of the session. We are currently having 100 plus participants on YouTube. So comments received on YouTube will also be typed in the chat box for the monitoring. Over to you, ma'am. Thank you.
Okay, thank you. It is a very proud moment for me to participate in this workshop and to introduce a past student of ours, Sanish Davis, who is currently the president of the Indian Society for Clinical Research and currently working as an R&D director of Janssen Pharmaceutical Companies. Dr. Davis has previously worked as head global clinical trials and senior medical director of Covance and deputy general manager at Glenmark. So Dr. Sanish, please go ahead with your talk. You can share your screen.
Thank you, ma'am, and thank you to the Department of Clinical Pharmacology for the opportunity. Let me share my screen. Okay, and if somebody can let me know if they can see my screen as well as hear me, okay?
Dr. Sanish, all good. Please go ahead.
Okay, thank you. So today, I will actually try to compress my presentation much further than I thought. Obviously, we need to be on time. So we are trying to kind of like forge into an area from what Dr. Kulkarni talked in detail about vaccines to immunotherapy. So it's like it's in the same biological area, but in a different realm altogether. So these are my standard disclaimers. And what I plan to cover is a little bit about introduction about immunotherapy agents. This is especially in oncology. This is becoming more and more common as the days and the months go by. But there are, of course, you could say toxicity concerns as well as safety concerns that are still there, even with an agent which is sometimes very specific. We'll also talk about early clinical development and how that links to the safety as well as efficacy of this compound. So just to kind of like give a spin to those pharmacology as well as other students who might be listening to this. And also a little bit about why sometimes the standard thinking process of phase 1 designs, etc., which ultimately leads to tolerability, safety, and other material that is brought out from these studies may not be exactly going to play the same way for immunotherapy agents. And linking from early clinical development to late stage, what are some of the operational considerations that we are seeing with regards to safety report? And then hopefully we can have some questions. So as many of you know, immunotherapy agents are kind of like evolving at a rapid pace in multiple cancers. Initially, it started with maybe myeloma or maybe even for melanoma. And then now we are kind of like moving into so many different areas. You have agents like immune checkpoint inhibitors, monoclonal antibodies, cancer vaccine, and now the CAR-T therapy. And India can also rightfully boast of its, you could say, elevation into that same league, because we have Indian organizations which are actually doing CAR-T, you could say, clinical studies, and also hopefully we should get these approved some years down the line. So that's how the area is, you could say, booming. But with that also comes, you could say, some of the very specific challenges associated with these compounds. So one is cytokine release syndrome. And if I was giving this lecture maybe 4 years back, or maybe before COVID, many would actually say that, oh, wow, this is such a big challenge. And how do we manage this? But post-COVID, everyone who's actually related to the medical field knows what cytokine release syndrome is and how to manage this with the free availability of tocilizumab in today's time. But nevertheless, CRS management in a clinical trial setting may sometimes be much, much more different than from a clinical practice setting. And together with neurotoxicity, which is called as the ICANN toxicity that is seen, these can sometimes get severe with newer and newer agents that are there. And hence prompt recognition and management with corticosteroids are important. And with more CAR-Ts actually coming on play, this also leads to an added level of complexity because these CAR-T agents, once you actually provide it into the body as a therapy, you cannot kind of like stop it, discontinue it, or you could say wait it out, right? So that adds a level of complexity with these kind of agents if you have a CRS or an ICANN which develops. There are also specific adverse events related to some very specific, you could say, antibody drug conjugates or other immunotherapy agents, but these are by far not so common in this thing. So from the preliminary toxicity considerations that we talked about to how do you actually develop these kind of agents and how do you actually have much more control over their tolerability as well as toxicity profile in early development. And this all kind of like is spurred by the breakthrough designation for experimental drugs that the US has brought out in 2012. What this states is that if you're actually, you could say, developing a drug for a serious or a life-threatening condition for which preliminary evidence shows that there is substantial improvement or existing therapy, then that is a drug which can get a breakthrough designation. Now, such designation accelerates the review of the compound. So when you actually submit for a clinical trial or when you actually submit for a new drug approval, there is significant acceleration of the review process by the US FDA. And also there is a huge amount of back and forth communication, guidance, etc., that is available from the agency on a standing basis, which means that the country can plug into the US FDA. Sometimes people say at will, because these are drugs which have the breakthrough designation that is there. So pembrolizumab or Keytruda, the drug from Merck, was the first one which kind of like got approved this through this pathway. And the approval was through a phase 1 program, right? I mean, this is how things are changing. So they actually looked at a multi-center open label randomized phase 1b study, because this used patients in majority of the cohorts. And the commitment that the FDA required is that the firm should conduct a phase 3 study with SOC, but we are giving you approval because this is a breakthrough designation. So that's how things are moving. So what this means is that there has to be much more robust definition of safety and tolerability in early phases before the drug can actually come into marketing approval. And because the data is going to be based on the phase 1, where the approval is going to be given, there is a much greater emphasis now on the, you could say, the clinical trial design and early clinical development. So the traditional approach to doing oncology dose finding, etc., is the standard phase 1 design, which is called as the 3+3 design, where you try to assess DLT and MTD. But for immunotherapy agents, these assumptions may not be exactly the same, and hence there will be challenges. So given the huge thrust that is given for the phase 1 data, which is leading to giving approvals for these compounds, there is a growing need for understanding how you can better define safety as well as better define efficacy and all that, and move forward for these new drugs that are there. So the standard way in which we have been doing oncology phase 1 studies or early clinical development has always been that we kind of like estimate the MTD, and then find the DLT, etc. But that is because we have always been thinking that if a drug is more toxic, then obviously it should also be more efficacious. And that is called as the monotonicity assumption or the monotonicity paradigm. Now, this paradigm is going to be completely overturned for immunotherapy agents, because the MTD and DLTs may not be very obvious for these kind of agents as compared to the standard small molecule chemotherapy that we are being used to. So the goal is shifting from goal 1 and 2 to goal 3, where you want to actually treat patients and find out what is the optimal biologically active dose level, right? And that also gives you a sense of the safety and tolerability of the compound. Now, this is something which everyone knows, and I think the red box is what is actually important, that when you have dose limiting toxicity, then you either escalate or de-escalate depending upon how many number of patients have the serious toxicity. That's it. So this is the standard 3+3 design, which we all know about. But there are certain very specific nuances for these immunotherapy agents which prevent assumptions from a standard 3+3 designs to actually not be possible to extrapolate. The first one is these agents have late onset toxicities. So traditionally in phase 1 studies or early development with chemotherapy drugs, the DLT or dose limiting toxicity was determined basically in cycle 1 of the treatment. But in immunotherapy agents, the cycle 1 is not the only one. I mean, sometimes you have to dose for several cycles before you actually get to what is called as the optimal biological effect. And hence, the standard assumptions of finding out DLTs and MTDs, or maximum tolerated dose, in cycle 1 may not be, you could say, be the most appropriate. So you need to kind of like see that when these immunotherapy agents are administered over extended period of time, they can also generate what is called as the immune-related adverse event, and that can sometimes occur sometimes in cycle 2 or cycle 3 even, right? But most probably people say that you can still see some of these in cycle 1. So what does that mean? It means that you have to actually sometimes also extend the DLT evaluation window from your just one cycle to maybe a couple of cycles. And you would also probably have to bring in newer assessment techniques for observing these DLTs in a much more micro fashion. So what they call as the time to even continual reassessment method, or the TIT CRM method, which is just a way to kind of like see that if you have even minor tolerability issues that you're seeing, you need to kind of like completely evaluate that patient in order to kind of like see if there is any trends or others that are there. So you can either extend the time period of evaluation from one cycle to maybe other cycles too, or within the same cycle or the first cycle, you might have to do a much more richer toxicity in assessment that is there. The second change that is coming about is the dichotomy from the traditional approach, which is saying that if you dose more, or if the toxicity is kind of like more, you will get better effects, right? Or the drug could be more efficacious. And you need to balance between toxicity and efficacy in order to actually go through with the dose that you want to select for the next phases. But immunology agents are not going to directly kill the malignant cells, right? They are going to have, you could say, this gamboling kind of effect where they will go and affect a T cell or a natural killer cells, and those indirectly facilitate the cytotoxic efficacy of these agents. So it's not a direct cell kill, but they kind of like affect the immunology cells in order to kind of like do this. So from MTD, we'll probably have to move into determining what is the minimum effective dose. And there have been several examples in the literature where you would see that the dose efficacy curve actually flattens out for these kind of compounds after a certain dose. So you can't actually say that you have reached an MTD because there are different of these definitions of the maximum tolerated dose. So that's why for several of these compounds, sometimes you cannot actually state the MTD, but you will have to actually go with a lower dose and find out what is the minimum effective dose. And we've linked this concept in the next slide with what we are trying to do, which is bringing the recommended dose that you want to actually do with your further clinical development. So you might have, you could say, determine what is your minimum dose, etc. But there should also be early on assessment that you can incorporate other endpoints like tumor activity endpoints. So you can use a lot of biomarkers, clinical assessment, pharmacokinetics, all these things, including persistent immune effects, etc., to ensure that you have these kind of endpoints that are there. And link these tumor activity endpoints to your minimum biologically effective dose. And that ultimately you would say is the biologically active dose that you're taking into your phase 2 and onward. So that's the linkage that you want to do. You just don't want to, you could say, restrict yourself to the MTDs and the dose limiting toxicity, but you're changing from all these traditional ones to the minimum biologically effective dose and incorporating other parameters like PK, PD, and all the others in order to get what you call as the optimal biologically active dose. And this would become your phase 2 starting dose. The fourth change is because of all these agents are going to be developed in combination. No longer will you have a single agent being developed and then later on you add to a combination. The current development paradigm is that you are starting with maybe a double drug combination, or sometimes even triple drug combination. So the challenge is that how do you ensure that these toxicities of these drugs are not kind of like becoming overlapping, right? So the paradigm is to kind of like change from just having one drug to actually having multiple drugs that you're going to test at the same time. So the time of monotherapy trials are kind of like slowly becoming past, and this will probably be replaced by combination therapy testing. And it's going to be more challenging because you can't find an MTD, because sometimes these two drugs might individually also have different MTDs, toxicities, and efficacy profiles, which might not be easy to understand. So what they're trying to do is identify non-overlapping toxicity profile drugs whenever possible. But since that is always going to be there, you will need to find out what is the best way to actually have toxicity profiling, even for those agents with newer markers, pharmacokinetics, and all the other immunology markers that are coming into being. So phase 1 drug development is not going to be very easy for combination therapy. You may not sometimes be able to do all this in 3 months, 4 months. You might sometimes have to have a longer window period to complete your phase 1 program. So from early clinical development and some of those challenges, let us look at safety reporting in late stage development. And before that, I think what is spurring all this and why is it important for India and for, you could say, clinicians and investigators from the perspective of, you could say, immuno-oncology. So oncology clinical development is like, it's like a 100-meter race, right? It's getting to the first place makes all the difference, because if you're second in line, there's hardly any sales that sometimes happens. Also, it's a big, you could say, slope to actually climb. And traditionally, only a limited set of countries are usually involved, and that's based on regulatory turnaround timing, site readiness, and all the others. And there is always an established old boys club within early clinical development. US, Japan, China, Spain, Poland, and some of our neighbors like Malaysia, Korea, etc. are also in this old boys club. So there is huge resistance to include countries like India. And obviously, because of all these other challenges that we have from a regulatory approval timeline, approval of amendments and all that stuff that is still there. But there is a recognition that India brings to the table a lot of, you could say, opportunity to actually get different safety tolerability information, right? I mean, 1.4 billion people is no, you could say, passover or no pushover here, right? You definitely can't actually ignore this huge population. So having appropriate safety reporting in early clinical trials is important, just like what I said earlier, that the breakthrough designation is making phase 1 study data all the more important, right? So how do you actually collect more information in the phase 1 studies and, of course, supplement it with data from phase 3 studies? And India is currently seeing a lot of phase 3 studies, but less of phase 1. So for our regulators also, it is important that the safety data that is collected in our phase 3 are going to be important from the assessment of how these drugs are behaving in the Indian population. And add to that, there are also these challenges which are more higher in India. Like we have a higher withdrawal of consent that happens in oncology studies. We have a larger number of lost to follow patients in our oncology studies. We also see some reports where sepsis is higher in Indian patients, and not just because of economic condition, but probably also because sometimes it is not well understood or well not treated, etc., or treated early, etc. So these challenges also add to the complexity of safety reporting and the importance of that. And it's important for our investigators as well as clinicians to have an open conversations with patients on the importance of safety reporting. Traditionally, I think clinicians would hesitate to talk more about safety because sometimes it's an admission of blame, of guilt, and all these stuff, right? So how do we break from that cultural barrier and have these conversations when the study is ongoing or when the treatment is ongoing? So I'll talk about a couple of scenarios that we have been observing in our clinical trials, and all of them are related to safety. The first one is on investigator judgment, right? Many of our Indian peers, Indian investigators feel that I know as much of oncology, my patients, my therapy area as a compatriot in the US or Europe or other places, right? That's a stated one. So here is a patient who has a solid tumor malignancy and is on existing chemotherapy, and an immunotherapy agent has been started as part of the study. So the patient is on 4 drugs simultaneously, 2 study drugs and 2 chemotherapy drugs. The patient develops neutropenia, and the investigator feels that it is definitely related to the drug. And then the question is on causality, right? Does the investigator feel that it is due to all 4 medication, or is it due to one medication or the other? So while the investigator has said it is yes, but there is no further information as to what agent does the investigator think it is due to, whether it is due to the study drug or due to the chemotherapy. Now, there is huge pushback from a global company because obviously they would want to ensure that their drugs are not under the scanner unnecessary, right? So when the global study physician asks for these additional data, the investigator just kind of, you could say, shuts down or says that, no, I don't want to discuss any further. I have reported my stuff. You guys can do what you want. I'm not bothered, right? So those are some of the kind of challenges that can come up. And we have heard these challenges happen in many of our studies, not just in one company, but across the ecosystem. But what can we do to help the PI from a safety perspective? One is, of course, the line listings and the information that is in the investigator brochure. If it is not there, then there should be an evaluation of other factors, including the data for existing chemotherapy. And that's an important thing which the investigator has to do. The CRA, the project manager, even the medical director has to actually get into this, because sometimes the investigator feels that I don't have time to get into all this, but there is a way out of it. So how do we actually get to it, and how do we make a more rational discussion? And it's always good to actually have a discussion with the medical monitor before causality decisions are made, because sometimes these are made in haste, and then the investigator has to retrace whatever has been done. And that creates a lot of issues from a ethics committee perspective, as well as from a regulatory perspective. The second thing that we are seeing, interestingly, is called as social media activism. So these are studies which are ongoing. People immediately put it on Twitter. The intent is to actually ask for more information, right? So this is an example of what information that is there, the details on this thing. And if you can see the Twitter handles, many of them are, you could say, experts that are there in the West or in the US. And the question is to see how to treat these kind of patients. But when this kind of like becomes a bit more difficult, the challenge is where the investigator in a clinical trial actually asks for opinion, right? How many of you think that we should actually go with this particular drug or another drug, or how do you think it is treatment is there? So investigators do this in good faith, but what happens is that sometimes you disclose the name of the drug, which is a breach of contract, and sometimes inadvertent safety issues are leaked into the social media. And that can actually have ramifications on additional patients who might be taking the drug, or sometimes patients might actually get into unnecessary, you could say, challenges with these things. And also, there are patient activism, legal implications, unblinding of the study drug, regulatory scrutiny, all this can come up. So what we have been telling investigators is that reach out responsibly. Know that you are working for the patient's benefit, but also ensure that safety is not something that can be given out in the public to gain opinion, right? Or gain acceptance. That has to be done within the confines of both a clinical research setting as well as in a clinical practice setting. Third is non-reporting of adverse events. So technically, I think this is something which everyone and Dr. Kulkarni has also stated about this, that sometimes there are adverse events which are very common, like for example, headache. Many of us feel that, and many don't even give that much attention. But in a clinical trial, especially with an immunotherapy agent or an immuno-oncology agent, it is very important that it has to be reported. Now, in one of our studies, we found during an audit that in the nursing notes that it was mentioned that the patient had headache, itching around the eye, etc. The investigator did not kind of like capture any of this in the source notes or in the medical notes. And when confronted, the investigator said that we don't actually give any credence to the nursing notes, and was very belligerent with regards to this. But to the auditor, it was very clear that these were important AEs that were missed because they were not taken cognizance of. So it is important to educate clinical trial sites on safety, how it needs to be reported. It needs to be factual in its reporting. And in a clinical trial, you don't have to fear about reporting, right? What is seen in post-marketing world also sometimes reflects in a clinical trial world. But that's an education that needs to be done for investigators and others. The fourth one is on serious adverse event reporting. So patient on a study had an SAE of intestinal bleeding with immuno-oncology drug. Reporting was all done appropriately. The sponsor reporting had to be done through what is called as a safety submission gateway, because that's the only way that it lands up with the safety physician for that particular study at a real-time basis. Unfortunately, the site, you could say, missed to report this even after 72 hours. And when they reported, the sponsor's company central team went into a tizzy because the safety physician is coming to know about this after several days of the actual event happening. So there are allegations that India doesn't report SAE properly, and then all those other, you could say, events which get triggered. So sometimes it's also important, I mean, for a sponsor to ensure that the safety thing is not just taken up as the last item on the investigator meeting or SIV. You need to constantly have this conversation with the site and others in order to do this. In today's time, you have virtual IMs and SIVs, so these issues might get even more compounded. And hence, the need for the CRA to ensure that the CRC at the site knows that every sponsor might have a different requirement. So you need to be very clear about what requirements are there from a safety perspective, right? So that's the important thing that is there. And the last one is, I think I'll end with one of the challenges with many ethics committees face. Oncology studies, and I'm sure many other studies also have huge amount of safety data that actually gets, you could say, thrown to the ethics committee. Sometimes the SUSAR listings or narrative listings can even be 100 or 150 pages, etc. And sites actually, when they receive it, just forwards it to the EC or the IRB. And some EC also takes up line listing only once in 3 months, which means that these are not discussed. And it's difficult as a sponsor to judge if these reports are discussed and whether it is documented in SA subcommittee meetings or others that are there. And ideally, the PI is supposed to review these SUSA listings and let the EC or the IRB know that, okay, is there an issue? Do we think there is a challenge, etc.? And there is a note to also to the sponsor that how do you actually make this much more engaging rather than just sending 100 pages listing to the ethics committee, because that gets lost in the whole discussion about safety and others with particular agent. So to come to an end, immunotherapy is seen in not only in cancer, but you have huge amount of research which is happening in all kinds of immunology conditions. So whether it is in cancer, whether it is in gastrointestinal disorders, whether it is in lupus, everywhere, I think there are so many conditions where immunotherapy is kind of like gaining ground. These agents have tremendous clinical activity across tumor types, but they also have different toxicity profiles and mechanism of action. The classical assumptions of chemotherapy agents may not be applicable for this, and hence there needs to be a very heightened awareness about safety reporting that needs to be done both in early stages and late stages. And that's the only way that we can ensure that within clinical trial setting as well as in the clinical research, in the clinical practice setting, the data that is kind of like brought out for a safety perspective and the patient safety are both of the highest order. So thank you so much once again for the opportunity, and look forward to seeing any questions if there are.
Thank you. Thank you, Sanish. That was very good. But I will go straight to the questions now. And there is one question at least. Given the design and development challenges outlined by you, how long do you continue pharmacovigilance in phase 1 studies?
Yeah. So the traditional, you could say, pharmacovigilance is 30 days post the last dose of the study drug, right? I mean, that's been the standard that is there. there are, of course, a lot of thoughts that you might need to have a longer period of, you could say, safety assessments for these compounds. But currently, I think we are still kind of like sticking to the regulations that are there. Hopefully, I think things might change. There could be a longer period of, you could say, safety assessments that can go on in these phase 1 studies.
Okay. I don't see any more questions here, so I guess that completes our session. Thank you very much, Sanish, for a very well discussed issue. And these immunotherapy agents are new, and I think we are evolving even the pharmacovigilance procedures or even the drug development procedures as we move along. So we have a lot, us also as a clinical pharmacology experts have a lot to learn. Thank you so much for your, I liked your comment about ethics committees, because this is what we used to face a lot during our time when I was working in the KEM ethics committee. I think it's something that industry also can do something about, perhaps evolve better reporting methodologies, and especially those SUSARs that come are impossible to evaluate. There are hundreds, as you said, and it is really difficult for the member secretary to make a presentation also to the rest of the committee, even the SAE subcommittee, to evaluate all these things. So thank you very much for making that point. Thank you. I think I will go back now to Nitya or Vijaya or Palvi it is.
Yes.
Okay.
Thank you, ma'am. And thank you, sir, for this session. So right now we have over 200 plus delegates with us on YouTube. So for the next session, I invite Dr. Jerin Cherian, Scientist D, Indian Council of Medical Research, Department of Health Research, New Delhi, as the chair of the session. With an MD in Pharmacology and Therapeutics and a Master's in Medical Law and Ethics, Dr. Jerin is currently working as a part of Division of Basic Medical Sciences, handling projects in the domains of essential medicines, rational use of medicines, and access to medicines. He is also a member of the national team developing standard treatment workflows for the National Healthcare Program ABPMGAY. We are grateful to him for taking out the time from his hectic schedule. I request Dr. Jerin to kindly introduce the speaker and take the session forward. We also request you to monitor the chat box for the discussion at the end of the session. Over to you, Dr. Jerin.
Thanks a lot. And thank you so much for this invitation. And it's an honor and pleasure to be here. This is a very interesting topic, and also a bit off the tangent from whatever has been discussed this far, because we had vaccines and then we had immunotherapy. And I think we are perhaps going in increasing levels of complexity, because immediately after material vigilance comes biosimilars. So the speaker for this is Dr. Melvin George, and he's been a close friend and colleague of mine. We've known each other from his JIPMER days, where he did his MD and his DM, and he's a clinical pharmacologist currently working as a professor in SRM Medical College in Chennai. He's been part of multiple international multi-centric clinical trials and also co-authored more than 90 peer publications in peer-reviewed journals. I've heard some of his talks in the past, and he's a very nice orator as well. So over to you, Dr. Melvin, and we are all listening to you.
Thank you, Dr. Jerin, for those words of introduction. I hope I'm audible.
Yes, sir. We can see your screen and you're audible, sir.
Okay, thank you. So at the outset, I wish to thank Dr. Nitya, ma'am, for giving me this opportunity to talk about this topic on medical devices. So if we look at the Indian scenario, the device market in India, as far as medical devices, it is estimated to be around $12 billion. And this is said to be a sunrise sector, which is growing. The annual growth rate is at 15%, almost, where at the same time, almost 75% of the market size is extensively dependent on imports. And there are almost 1,000 manufacturing sites in India for medical devices and 200 sites for in vitro diagnostic kits. And medical device industry in India is said to be the 4th largest in Asia after Japan, Korea, China. And worldwide, if you see, it's among the top 30. So we are quite far behind the other countries when it comes to the market size, but we have tremendous potential. And this is one sector where the government has allowed 100% foreign direct investment. And the price control as far as medical devices, to a large extent, it is not as tightly regulated as what we see with drugs, except maybe in a few instances like cardiac stents and knee implants. So medical devices, if you see, they have been, it's one area that has been largely unregulated, I would have to say. And for a long time, it was considered as medical devices were governed just under the ambit of Drugs and Cosmetic Act 1940, and they were looked on as drugs only. They were regulated as drugs itself. So there was no separate regulations for medical devices. So it was the Drugs and Cosmetic Act 1940 and Drugs and Cosmetic Rules 1945 for as long as 70 years. And then in 2017, we had a very major amendment to this situation, and that is the Medical Device Rules 2017. And subsequently, there was the Medical Device Amendment Rules to 2020. And then most recently, just a month back, we had the Medical Device Policy 2023. So prior to 2018, if you see, medical devices were just regulated under this Drugs and Cosmetic Rules. And under the definition of drug, it would say that devices are considered as those which are intended for internal or external use in the diagnosis, treatment, mitigation, or prevention of disease or disorder in human beings or animals, as may be specified from time to time with the Government of India. So, and if you look at the situation in the 80s, there was something called as notified medical devices. So certain devices alone were notified as drugs, means only they would be regulated. It started with syringes, needles, perfusion sets. Subsequently, that list was more devices came into that list: cardiac stents, drug-eluting stents, catheters, intraocular lenses, cannulas, bone cements, heart valves, scalp vein sets, orthopedic implants, internal prosthetic replacement. So as you can see, we have so many medical devices, but it is only the small subset of devices which are practically being regulated as late as even 2005. All other devices which were not under this category were totally unregulated. And then we had in 2017 this medical device rules coming into existence, and where they had listed down different devices which would be considered as notified and not notified devices. In that list, if you look at it, we had 37 categories of devices which were considered as regulated devices. And subsequently, we saw that in 2020, the rules changed where the concept of notified and non-notified devices totally discontinued. And we had a situation where all devices had to be regulated, had to be registered. So if you look at the medical devices rules 2017, it categorizes the risk classification of medical devices as low risk, that is class A, low to moderate risk, class B, moderate to high risk, class C, and high risk, class D. So this is a classification that is determined by the Office of the DCGI. So if you look at the website of DCGI, the Sugam portal, you will be able to find for each medical devices that is approved by the regulator what particular class it comes under. The reason is this is important is because the class A and class B devices are largely regulated by the state licensing authority, whereas class C and D are more directly regulated by the central central devices regulatory authority. And this, we have similar kind of classifications even in other countries like in the Europe, US, and Singapore, where we have, based on the risk, the devices are classified. And the standard of medical devices we have is based, the standard that is laid down is the Bureau of Indian Standards. And suppose if such standards are not available for a device, then we go by the pharmacopoeial standard or ISO standards. And just as how we have a recall of drugs, we also have the situation where a medical device needs to be recalled if a manufacturer or an agent who is supplying the medical device finds that there is a safety issue with the medical device. And it's supposed to be immediately recalled from the market. We also have a quality management system, which is called as ISO 13485. As of now, it's a voluntary standard that is laid down by ISO. It came out in '96, and it contains a comprehensive quality management system for design and manufacture of medical devices. So the challenge we have had, because medical devices were not very tightly regulated, we had a lot of medical devices in the country which are of substandard quality, and there was this challenge because they're not having such high standards. The prices would be highly competitive. As a result of that, they would be entering in the market without any standards, and that is when the Quality Council of India and the Association of Indian Manufacturers of Medical Devices launched the Indian Certification of Medical Devices 13485 plus scheme to undertake verification of quality, safety, and efficacy of medical devices, so that there is some kind of quality process that is ensured in the manufacture as well as in the quality, safety, and efficacy of medical devices. So in 2020, when the medical devices amendment rules came about, they practically changed the way devices are regulated in the country, because they said basically all devices, including an instrument, apparatus, appliance, implant, material, or any other article which is used alone or in combination, including software or accessories, which is primarily achieving its desired action not by a pharmacological or immunological or metabolic means, and it is used for diagnosis, prevention, monitoring, treatment of disease, disorder, or injury, disability, or investigation, replacement, modification of a physiological process, or sustaining life, or disinfection of medical device, or control of consumption. So this was the latest definition of device. So any of this, if a product is fulfilling this criteria, it would be considered as a medical device, and all such devices would have to register themselves in the online portal set up by CDSCO, and they had given them a voluntary basis time period of 8 months for registration. And for class A and class B devices, the deadline they had set up is for registration to obtain license. It was August of 2022. And for class C and D devices, they had given a little more time till August 2023. And even the supply chain of these previously unregulated class A, class B, as well as C and D devices would have to obtain a license from the regulator. So through this law, what has happened is practically every device in the country would have to go through the registration process and have some kind of registration process. So next we talk about clinical trial situation in India. When do we need to do clinical trials? So we start with when you're having a clinical trial, when you're having a medical device. Now, if it is a new device, there is no predicate device available already in the market, you obviously will be starting with preclinical research activities, which will include bench testing, technical testing, probably if there is possibility of doing computer simulations, that would also be done. And if it is relevant, even animal studies would be considered. So bench testing is essentially the lab testing or workshop testing that is done to assess the device performance and safety, and to ensure that the particular medical device that you're using is really designed to follow its particular function. Then we have the quality and engineering testing of the materials that are used. Probably different materials would be used to see which is the most optimal material for giving the accurate results and for ensuring the reliability of the device. And the prototype is usually built with different materials, and the best material is finally chosen during this phase. Then we also try to use computer simulations which can understand human biology and disease, by which you could fine-tune your device so that we can, based on this computer modeling, we can predict which would be the ideal setup to for the device to achieve the intended effect in either diagnosis or treatment in human disease. And efficacy testing, it is not recommended that it should be done for all devices, but if it is possible, it could be attempted because there is obviously a lot of anatomical biological differences for the device to be tested in animals. So it cannot be possible at every situation, but it is encouraged. Now, when we talk about clinical trials, we do not, in devices, we do not use phase 1, phase 2, phase 3. Rather, we use the word pilot study or feasibility studies and pivotal studies. So pilot studies are essentially studies done in a smaller group of individuals to understand how the device is working in humans. It's more an exploratory kind of study to collect preliminary safety data about the device performance in humans. Now, based on the results of this pilot study, you would have to come back to the drawing board and modify the device, and also the data will help us to design the pivotal study. And you could also explore the eligibility criteria and the practical application of the device in the pivotal clinical trial, which is the confirmatory trial or the equivalent of the phase 3 trial. And this traditional feasibility studies would be done in a slightly larger group of individuals just prior to the phase, that is the pivotal studies, where the object is to use the final device design, because in the pilot study, you're just going to use a device which may not be the ideal model. So you might have to change that and again test it in a smaller subset of individuals, maybe 20 to 30, before you go to the pivotal study. So that is what is done, where again safety is tested here and effectiveness is also tested. And then we come to the definitive or confirmatory studies, where numbers could vary between 100 to 300, where you're gathering evidence of the safety of the device. For medical device which doesn't have a predicate medical device but approved outside India, pivotal studies need to be carried out. So prior to conducting a pivotal clinical study in Indian subjects, sometimes the CDSO might say to do a pilot study first to see if the data that we are generating in India is confirmatory to what is already generated outside the country. So it's not that every time a device company could directly go for a pivotal study. It might be asked to do a pilot study also. And once this process is over and the product is approved in the market, and the efficacy has got good efficacy and safety results, and it is approved for use in the market, the regulator would obviously ask the sponsor to continue doing post-marketing clinical investigation, which would obviously be done in thousands of participants to assess the long-term effectiveness, safety, and usage of the device in the general population. And this would help the regulator as well as even the manufacturer for that matter to understand the optimum use of the intended device, because you're doing in a much larger population. Even safety would be known much better at this point, because as you can see in pivotal studies, you're not having very huge numbers when it comes to medical device trials. Many times the number is much less than 1,000. So you are unlikely to have rare adverse events happening in this pivotal trials. And so post-marketing clinical investigation is a very important component to understand the material vigilance of these devices. So there are some initiatives that are brought out by the government of India, the Make in India initiative, Atmanirbhar Bharat Abhiyan scheme, to improve this manufacturing of medical devices in the country. For instance, there is the production-linked incentive scheme for promoting domestic manufacturing of medical devices. So this is called PLI scheme. This is not only there for devices, it is there for many other sectors, telecom, many other sectors are there. And it is essentially a scheme that is promoting the companies to develop, to get incentive from the government based on the incremental sales of the medical devices that are manufactured in India. So that based on that, already there are several companies which are manufacturing devices like linear accelerator, cobalt machine, even blood pressure monitor, CT scan, MRI, all these devices. There are some of the companies I've just shown you, which have already obtained this PLI scheme based incentives. So the point I'm trying to say is government is trying to ensure that more of medical device manufacturing happens within the country, instead of we relying more on importing of medical devices, which obviously increases the cost to the end user, which is the patient. As we know, in India, majority of the hospital expenditure is borne by the patient himself. So if you're going to have the use of costly equipment, that is obviously going to increase the price that the patient is going to pay. And then the second aspect is development of medical device parks. So it is not easy to test medical devices in India because of the reason that the setup that is required to test a medical device is obviously a quite something that it will not be easy for every institution, every small startup company also to have such a massive infrastructure. So in order to have some kind of a common ecosystem where every startup company or even an academic investigator could use a common facility, this scheme has come up where a medical device park will be set up, and that medical device park will be available for use by different startup companies and medical device companies, as well as academic institutions, so that they could use this medical device parks to develop their product. So the based on this, it was an open call for different state governments, and four states were able to, in principle, get the nod for getting this medical device parks developed. We have one in Madhya Pradesh, then in Tamil Nadu, in Himachal Pradesh, and Uttar Pradesh. So the medical device parks are coming up. It's not yet, it's as of now, it's approved in principle. It's not yet come up, but we can see maybe in the next 2 or 3 years, this would be totally functional. And that would in turn help more number of medical devices to come up in the country, which are indigenously tested for its initial quality design, as well as the efficacy and safety subsequently. And lastly, we talk about the National Medical Device Policy 2023. It's a very forward-looking document that was just released last month, which aims to accelerate the medical device industry and also have a more patient-centric approach to meet the evolving healthcare needs of the patients in our country, and more importantly, to increase the India's share in the global market of medical devices to 10 to 12. Right now, it's less than 5%. So we are trying to see that this can be substantively increased. So some of the major aspects of this policy include access and universality for obtaining access to medical devices, and to develop more affordable medical devices, and to improve the quality of medical devices, and to ensure that there is more patient-centered and quality care through development of medical devices, and also to develop devices for preventive and promotive health. Now, this is something that is what we are seeing increasingly of late, is to develop medical devices which are able to predict the development of diseases much ahead of time. There are, for example, in the market right now, there are some devices which are looking at developing and screening for pre-diabetes, screening for hypertension, even much before the patient can possibly have clinically diagnosed them as hypertensive or diabetic. So these kind of devices are slowly coming up in the market, so that we are able to prevent diseases. And skilled manpower, obviously, if you're going to develop medical devices indigenously, it requires a lot of competence. So developing skilled manpower for that and ensuring this security exists for all the medical device companies, and to improve research and innovation. So under this medical device policy, it is expected that there would be regulatory streamlining. So one of the challenges that happens in Indian system is that at the moment, there is no single window clearance system available for approval of medical devices. So it's a very complicated system, which sometimes certain companies would not be wanting to go through that system of making their product available in the market, because it is too complicated, and many times it's not very explicit as to what is the exact requirement that the regulator has. So it's all based on how they read the guideline, and there's a lot of ambiguity in the guideline. So to avoid all this, if we can streamline and make it as a single window clearance system, so that it will be much easier for manufacturers to know what the regulators want and get their products approved. Enabling infrastructure, as I already told you, part of it was the development of medical device parks, and also developed systems in place where academic institutions, see, as of now, the system is such that an institution like NIPER or an ICMR institute and a few centers in India are able to venture into medical device research. But a lot of institutions are there where there are people who are doing good work, but then they do not have the facilities and the capacity building process not there in place. Infrastructure is not there in place for them to really scale up their design process. So this medical device policy is trying to ensure that the R&D and innovation, as well as the infrastructure requirement, would be increased, so that there could be participation of all these stakeholders. And of course, we all know, without investment, nothing is going to happen of great value. So attracting investments, especially involving getting funding from venture capitalists, human resources development, is something that the policy is trying to give a lot of importance. Essentially, using Skill India initiative, there is new policies in place to develop manpower that can really have the wherewithal within to develop medical devices within the country. As of now, there are a lot of sectors for which in India there is simply no expertise available to bring out such devices, and so we are bound to go for imports. So to change that, so that we have in-house capabilities within the country to develop which can either upskill people and help people to develop medical devices, especially those which are of complex nature. And of course, to build brand positioning of the Indian medical device industry, so that we are able to attract more foreign players to import our medical devices, and so thereby the global medical device brand of India thrives. So all this is going to be done by having some quality metrics in place. So accessing and affordability of safe medical device, how much is this achieved? Self-reliance in manufacturing, how much is this achieved? Improvement in overall health index and reduced disease burden in India as a result of all these processes, how much has happened? And finally, does all this lead to creation of high value in the jobs in the R&D and innovation sector? So this is how the government is going to track the outcome of this medical device policy. And nevertheless, there are a lot of challenges in the medical devices sector. One is the manufacturers have to navigate the inconsistent regulations. Many times it's all how the wordings are interpreted. So there can be a situation where things are not as clear as what it should be. And as a result of that, it stalls down the time taken for the manufacturer to get the approval. And adoption of cutting edge technologies, as I told you, in certain fields, for example, AI, cloud computing, robotics, all this is highly limited in Indian medical device sector. So we are bound to, at the moment, rely on Western imports for getting these kind of devices. So this should change so that we are able to improve our presence in the medical device sector. As I told you, and India also depends heavily on imports of medical device, leading to high import bills, thereby adding to the cost of healthcare. This is what I told you earlier. So if we are able to really make this change where we have our own medical devices, it would significantly reduce the cost of healthcare. And access, providing a system by which we have access to funding is also a critical challenge, because there is a reluctance on many investors, venture capitalists, to fund medical devices, because this sector has a lot of regulatory uncertainties and a longer gestation period than many other sectors. So we are hoping that medical devices policy 2023 would change all that in the near future. And coming to Material Vigilance Program of India, this is something that was established in 2015. Good thing is this is even there, because there are many countries which don't even have a Material Vigilance Program of India. So this is formally launched by IPC Ghaziabad. They are the ones who are just like how we have drug pharmacovigilance. So this is mainly monitored by IPC Ghaziabad. And the Central Coordinating Center for Material Vigilance Program of India, the National Coordinating Center, is Sri Chitra Tirunal Institute of Medical Sciences and Technology in Trivandrum. So they are the apex center. So essentially what happens is that all the reports of medical device safety issues from different healthcare professionals, manufacturers, importers would be reaching this monitoring centers, medical device monitoring centers. There are as many as on date, there are as many as 293 medical devices monitoring centers in India. So they get the reports of these medical device issues, and that data is finally reaching IPC Ghaziabad, and that data is being monitored by the Sri Chitra Tirunal Institute, as well as the Technical and Resource Center also provides their inputs to IPC in Ghaziabad. And we are ultimately, based on the inputs received, the decision to recall devices is also taken finally by the CDSCO in New Delhi. So for in the interest of time, we will close, and I'll be happy to take questions here. So to close, I would say medical device regulations in India have undergone substantial changes for the better, especially in the last 5 years. And the major change that has happened is the fact that now almost every medical device is being regulated. And the medical device policy implementation in 2023 would obviously alter the size and quality of medical device market in India. At least that much is envisaged. And we hope to see that happening really maybe in 5 to 10 years. Thank you.
Thanks a lot, Dr. Melvin, for that elaborate presentation. And you covered both the development aspects of medical devices as well as the vigilance aspect. So that was quite exhaustive. So just to go back to what he was saying, Dr. Melvin had actually mentioned that a lot of the high value medical devices are being imported, and that's the reason why the medical device policy in India is put in place to try and nudge both investors as well as investigators to develop indigenous products. And in that line, we have maybe one very important question, and I know that you did touch upon it, but maybe you can once again briefly summarize to our audience exactly what would be the clinical trial pathways and differentiated based on the risk classification. So as an example, perhaps you could take a device which is in a class B, which is of relatively low risk, and maybe a class D device. So could you please explain what would be the clinical development pathways for these two?
Yeah. Thank you, Dr. Jaren. So when we talk about the necessity of doing a clinical trial, if it is a class A or a class B device, the pathway for that to get approval would be to submit their process details of how they are manufacturing, about the details of the device to the regulatory agency, and they would only have to show data about the quality systems that they have in place for manufacture. There would not be any need for them to provide data on clinical use of the medical device. So if they are able to show that they have some quality process in place to ensure that the product that they have manufacturing is of sufficient quality, that would be sufficient for the regulator to give them the license. And once, of course, all devices, be it class A, B, C, D, ultimately you can find the information about that device in the Sugam portal. It would show up saying that this particular device has been approved for this particular indication from this particular organization, and it is a class A or class B or class C or class D. But the direct regulation is largely by the state medical licensing authority. So they would be the ones who will be doing the inspection of the site. Suppose if there is if they want to check whether this particular organization has adequate manufacturing capabilities to bring out this product, they would be the ones who are looking at that aspect. So to a large extent, A and B devices without trials are going to be regulated by the state medical licensing authority. When it comes to class C, class D devices, because they are of high risk, they are largely regulated by the central licensing authority. Now, when it comes to class C, class D, if it is a device for which there is already a predicate device available in India, meaning a device that is already in India, it is in the market for the same indication, and there is just another product from a different organization, a different device agency, then there would not be a requirement to carry out a clinical trial with the device. But if there is no predicate device available in the country, and this is the first device that is of this kind in the country, there is then going to be a mandate to do a clinical investigation, that is to do a clinical trial and provide data. And they can say, start with pivotal pilot study, show us the results. If you are convinced, then we will ask you to do a full-fledged pivotal study. Or they could say, go ahead with a pivotal study, demonstrate the efficacy and safety, and if you are convinced, we will give you the license to market your product. So that is how it is largely the pathway for medical devices in the country. So class C and D, there could be request from the CDSU to do clinical investigation and prove efficacy and safety, whereas for class A and B, that is not compelling on them.
And also the onus of establishing the defenses to state that there is a predicate lies on the developer or the investigator.
Yes. So they have to show that their device is not substantially different from the predicate device that is existing in the market, and because of which there is no need to really do a clinical investigation through a clinical trial. Yes.
Thanks, Dr. Nath. There was just one last question, and that was, and I think it's also a very pertinent question, because many of us do walk around with devices these days that have health components built into it, such as smartwatches, which nowadays have built-in ECGs, SpO2s. Some of them even send out alarms based on, and we may have read the news where there was a cyclist who fell off from his cycle into a ditch, and it was because of the alert that was sent out by his watch automatically that help could arrive in time. So I think this is an important question, and they want to ask, what is the regulatory status of such devices, such as those wearable devices that we use, which plot ECG graphs and things like that?
Yeah. So to the best of my understanding, when it comes to medical devices, we are looking at two different things here. The medical devices that are primarily used by a healthcare professional in a hospital setting, where those are the kind of devices which are at the moment being regulated stringently by these laws. So to the best of my understanding, the other devices, which are, for example, even the watches that we use, which are providing some kind of health data, they are not being treated as medical devices, because at the end of the day, that data is not being used by a healthcare professional to monitor the health or to diagnose a disease or to treat a disease. So they are not that much regulated, is what I understand, by the medical device rules and medical device regulations. Maybe that situation may change, but I think with the amount of applications that Sugam portal, I mean the CDSU receives to regulate the regular medical devices, I think this would take a much more time for them to come to this level to start regulating even these simpler devices.
Correct. I believe so too, Dr. Melvin. And if you would look at the definition of medical devices on USFDA as well as CDSU, they are more or less aligned. And they classify any device which are required for, like you said, diagnosis, prognostication, therapeutic implications. But the WHO definition for medical devices is a slightly broader definition, which includes promoting healthier populations as well. So yeah, there is that discussion still happening, I believe.
Yes.
So thank you so much, Dr.
In fact, there are a lot of devices that are coming up these days, which are targeting people going to the gyms, which are targeting people who are interested to know about their health much before they even develop a disease. So I understand FDA is tightly regulating even these kind of devices, but maybe in India it might take some more time for them to reach that level where they start regulating these kind of devices. Yeah.
Thank you so much. So thank you. Over to you, Nithya ma'am.
Thank you, Dr. Jairan and Dr. Melvin. With this, we come to the last session for the day. I request Dr. Nithya Gupte ma'am to kindly chair the session and introduce the speaker. I also request ma'am to kindly monitor the chat box for discussion at the end of the session.
Good afternoon, everybody. And Dr. Abhi, Dr. Sameer is not able to be here with us today. He got called away for an institutional commitment. He has told me that you all were together and he was actually looking forward to listening to you and sharing the session, but he's not here with us. So let me introduce Dr. Abir Saraswat, whom I had the privilege of listening to some weeks ago. Dr. Saraswat is a consultant dermatologist based out of Lucknow who works at the Industry Skin Clinic. He has completed his undergraduate education from the Armed Forces Medical College, Pune, and post-graduation from the PGI Chandigarh. He's an outstanding orator, I can tell you that, and I'm sure he'll enthrall you with his talk on biosimilars, the Indian scene, how similar, how safe. Over to you, Dr. Saraswat. Sorry for the delay, and thank you for accepting our invitation. Over to you.
It's okay. Thank you. It's a great pleasure. For a change to talk to a different audience. I usually talk to dermatologists. I don't know how well I'll be able to adapt my talk to this audience, but I'll try. I'll just start sharing my screen. Give me a sec, I'll just. So this talk is going to be from the perspective of a dermatologist, and I'm going to talk about the biosimilar scene in India from the perspective of mainly dermatologic biosimilars, because that's what I am exposed to, and that is what I have sort of experience of. And this I realized as the course of preparation of this talk that, just give me a sec.
We've made you co-host, Dr. Abira. You should be able to share screen here.
I'll just, can you see my screen now?
Yes. Perfect.
All right, I'll just make it full screen.
Yeah, and your voice quality also good. Yeah, please go ahead.
Yeah, just a second. There is a menu blocking. Okay, if you don't mind, I can continue like this because there is a
Your cursor is
menu
on.
bar blocking me, preventing me from making it full screen.
Yeah.
Okay, I think I can do it now. Yeah. All right.
Yeah.
So Indian biosimilars are a category that is relatively new. Biosimilars itself is a category that's new, biologics rather. And the last 20 or 30 years, there has been an explosion of these drugs, which are basically, I don't need to tell a pharmacology audience what biologics are, but this talk was made for dermatologists mainly. So I thought I'll just talk about, because many times when we talk about biosimilars, the picture in mind that appears is that they are the same as, branded versus generics. So basically a biosimilar is a generic version of a biologic that was developed by somebody. This could not be farther from the truth. And this is something important to understand, because only then we'll understand what biosimilars are, how they differ, what differences and possible dangers they may have, which the originator may not have. So this is just an illustrative example that shows you how complicated proteins are. Sometimes we forget. So paracetamol is this simple little structure. We have tons of generics of the original paracetamol. Premilast is a newly developed phosphodiesterase inhibitor, slightly more complex. Compared to this, I mean, we can't even show a protein at this scale. So, it is several thousand times larger. You can see the crystal structure of the IgG antibody here. So these are much, much more complex drugs, much, much more complex proteins. And the complexity is not just in the size, but it's in the way that they are in their natural state. So they have a primary structure, which, as you can see, is just chain of amino acids. And then they are linked to each other in various ways by various types of bonds. Then they twist and turn and form pleats and helices. And then they are post-translationally modified. So this just shows, this cartoon shows it nicely. A primary structure is just a chain of amino acids. Then there's a secondary structure. There are hydrogen bonds forming either a pleated sheet or a alpha helix. Then there's a tertiary structure where there may be a single or multiple subunits. The proteins fold and form various grooves and niches within which there might be receptors and domains where active sites may be there for binding of various other molecules. And of course, there is a quaternary structure where there might be multiple subunits. So it's a hugely complex thing. And this is just, again, a tabular representation of what all the body does to a protein to make it different from another. So there can be addition of chemical groups, various chemical groups like phosphates, acetylates, etc. can be added. There can be glycosylation, AMP can be added, lipids can be attached to them. There can be ubiquination. They can be cleaved in various different ways from various sites. And of course, amino acid modification also can occur. So huge complexity in proteins. That is why biologics are so difficult to duplicate or to make copies of using slightly different methods, or if we try to deviate from the proprietary method based on which the originator made them and did the original trials and found them effective for a certain condition. So, and there are various sources of these differences. As you can see, there are unique cell lines used by different manufacturers to make the basic, the building blocks of the biologic. And then there are proprietary process and growth conditions, which are often secret. Manufacturing process itself can change with time. For example, a very prominent biologic that we use, that is adalimumab, the originator himself, a few years back, they themselves changed the cell line on which they were making it. And they had to do extensive testing to show that it was the same as the previous one. Purification levels and methods are variable. For example, if just the immunoglobulin starts to clump together, it can become immunogenic. It can be the same as another immunoglobulin in every other way, but if it tends to clump together because of various reasons, maybe pH or electrical charges, they can become immunogenic. No two batches are identical, even of the originator. So, I mean, they need extensive testing, again, much more than ordinary drugs, I would assume. And of course, tertiary and quaternary structure is almost impossible to replicate 100% in copies. So this just gives you an example of the complexity involved in copying a biologic. So again, so this is just a tabular representation of the same thing. You could choose a particular gene sequence, then you could choose various different vectors in which to express it, make a plasmid, then infect a host cell. You could choose mammalian, plant versus bacterium for the same thing, and the end result would be slightly different. You could use a different cell line or a growth medium or an expansion method to expand it. And then you could purify it in various different ways to various degrees to make the final biological product. So each of these 8 or 10 steps, if one is slightly different, you will not have exactly the same product. So now that we've understood the complexity involved in making biologics and biosimilars, let's have some definitions. So a biosimilar is a definition that is clearly defined by the World Health Organization. So this is basically a follow-on biologic, which is approved by a multi-step biosimilar regulatory pathway pioneered by the European Medicines Agency and subsequently accepted as the standard by the WHO. So if you go by this definition, there are only a very, very few proper biosimilars in India that qualify, because most brands have not sought approval from the European Medicine Agency or the US FDA and have not qualified those standards which are required there. Therefore, they don't qualify to be called as such. So that is why the Indian pharmacology industry or the pharmaceutical industry has, or the pharmaceutical administration has developed a term called similar biologics. So these are the biologics that are copied by someone else and they are approved by the Indian regulatory pathway, which is not the same as the EMA WHO pathway. And there's another term that is called intended copy biologics, which is another way of saying the same thing, also called biomimics or biocomparable biologics, which is used to describe various off-patent biologics marketed in many developing countries like India, whose regulatory processes are not as comprehensive or as strict as the originators who coined the term biosimilar. So these Indian similar biologics have evolved through a regulatory pathway because in the early 2000s or 2010s, when they started coming in, and I think erythropoietin was the first one that came into the Indian market, there were no clear standards set for biologic approval in India. And approval was given on a case-by-case basis, ad hoc, and there was a very short abbreviated procedure, and it was done to the people who applied. On 15 September, these standards were sort of codified, and the standard was set, and a definition was made, which was that a similar biologic product in India is that which is similar in terms of quality, safety, and efficacy to an approved reference biological product based on comparability. Now, again, this is not a very, very precise definition. Comparability defined how, quality, safety, so many things are left unsaid in this. So these standards, again, in 2016 were made somewhat more stringent because probably the regulator realized that these were not good enough, and probably a lot of non-similar biologics or not to those standards were getting approved in India. And this is just an extract from the guidelines on similar biologics from the CDSCO, which was the rules that was till 2012. So just a structural and functional comparability had to be characterized in vitro, and it had to have all preclinical evaluations conducted. Pharmacokinetic and pharmacodynamic studies had to show that they were largely similar. Immunological assessment was essential because these can be potentially immunogenic drugs, and a comprehensive post-marketing sort of surveillance had to be decided. As you can see, the glaring omission here is actual human clinical trials. So they were being approved without that. So the 2016 modifications did bring in that. And they said that the clinical efficacy study cannot be waived, especially for large molecular biologics like monoclonal antibodies, which is what most therapeutic biologics that we use, at least in dermatology, are. But again, there were various riders in our rules. So here they say this, and then they say that it can be waived if all the conditions approved are granted: comparable quality, convincing PK/PD data. Again, they said confirmatory clinical study and efficacy study cannot be waived if there is no reliable pharmacodynamic marker validated. But again, they mitigate this stipulation also by saying that if there is a surrogate marker of efficacy, they can sort of waive it off in various ways. So this is just to show you that our rules are still not as stringent as rules were elsewhere in the world, especially even though these drugs are tremendously expensive, are given in some life-threatening and very serious conditions, and can have potentially problematic side effects if they are not what we think they are, or what they're supposed to be, which is a very, very close copy for all intents and purposes, which is supposed to have the same clinical effect as the originator. And of course, the post-marketing surveillance, which is the duty of every manufacturer and is codified for biologics, like for all other new drugs, is also left to the manufacturers. And myself, being a clinician, I can tell you that this is something, that is done in a very lackluster, lackadaisical manner. And at least I have not been approached by any pharmaceutical company except for one to submit post-marketing data on a newly introduced biologic in India. So this is just a list of some prominently used biologics in dermatology that were approved before August 2016. And only the two that I've marked in red are the ones that probably or certainly comply by the EMA WHO standards and probably can be called proper biosimilars. All the others are Indian biomimics or similar biologics, whichever you want to call them. And we don't really know whether they are actually similar or not. And consequently, we don't know how safe they are. Are they as safe as the originator? And the various problems that have been identified, this is not just a theoretical discussion. So this is something that a lot of people have listed. There is paucity of data in terms of these intended biosimilars in India in all aspects of product development, manufacturer, control of drug supervision, etc. Many times the different product chosen is not justified, isolation process is not described, there are no or minimal comparability exercises, the description of purification is not as detailed, investigation of impurities is insufficient, batch-to-batch consistency not demonstrated, validation of various infectious agents, etc. is not provided. So all in all, there is inadequate testing and non-orthogonal approach, and which leaves in mind a lot of uncertainty as to how similar and safe they are. And again, physicochemical or biochemical characteristics also, there's very limited data that is asked for by the government, not as comprehensive as in other countries and other continents. And that's how it is provided. Stability studies are also limited. Non-clinical studies are very minimalistic. Bare minimum number are done. I'll give you an example shortly. And stability profiles are not very well established. Adequate clinical data, poor study designs, the usual problems with the Indian pharmaceutical research are present here as well. Immunogenicity is also not very well investigated in many products. So these are not the criticisms of any single biologic. These are problems that have been identified across a lot of broad range of different biologics. And prescribing information and labeling is also very minimal. Requirements in other countries are also very strict, which I'll also talk about shortly, and which can produce and does produce a lot of confusion in the mind of the consumer. So again, now that in 2016, these criteria were tightened, are they the same as the benchmarks? Like I said, no, they are still not. And this is a prominent example. There was a trastuzumab biosimilar that was approved by the EMA. The same manufacturer did a study with 640 patients and got approval in Europe, whereas they just did a study of 100 patients in India, and they were allowed to do it. And of course, post-marketing studies in pharmacovigilance in India is subpar. We know that. And of course, a lot of discretionary power is still there with CDSCO to waive or dilute these requirements that have been codified, which ideally should be minimized. So again, this approach is not all bad. There are pros and cons of this approach, and I'm sure our administrators in their wisdom has chosen this approach for a reason. And the biggest advantage is that we have some of the cheapest similar biologics anywhere in the world available to the clinicians in India. And this has improved access to these biologics in a big way. Although still they continue to be quite expensive, but they are comparably much cheaper compared to developed countries. The cons are, of course, that there is a possibility of higher or new unknown adverse effects. There's possibility of poorer efficacy. There is a very distinct possibility of higher immunogenicity because these are all proteins which can be immunogenic. And brand switching carries a higher risk. In India, we know that patients tend to shop pharmaceuticals and pharmacists play fast and loose with brands. They themselves give their advice to patients. Why don't you buy this other brand? It is slightly cheaper. It's the same thing. And they think, it's like a generic thing or a different brand. Whereas, as my preceding slides have shown, a similar biologic is a very different beast from a generic copy of a branded small molecule drug. And so I end with a little bit of clinical experience that we've had with these similar biologics. So again, like I said, there are two biologics that I can quote from my specialty. One is an anti-TNF, the other is an anti-CD220 antibody, for which there is good quality data. And we know that with some confidence that they are actual biosimilars. For all other brands, there are very small retrospective studies, case series. There's no good quality data versus the innovator available. So we don't know. And this kind of testing has been done. This was a study published last year. And there were significant differences in lab assessments versus clinical assessment of 3 Indian similar biologics of rituximab. Now, rituximab is used to treat severe life-threatening conditions like pemphigus, lymphomas, leukemias, and we want a very high degree of certainty in this particular type of drug. But you can see that there were significant variations in the laboratory comparisons, and of course, but the biological activities seem to be pretty similar. But this was based on these basic assays: antibody-dependent cell-mediated cytotoxicity, or the complement-dependent cytotoxicity, or the flow cytometer cell sorting. But again, you never know unless you do large studies, you don't know whether these translate into differences in actual clinical outcomes and side effects. So this is a huge unknown, which then seems to be dumped onto the clinician who gets these products in their hands. And largely then we are treating our patients as study subjects, because these are inadequately studied drugs, at least by the standards of the world, of the developed world. And then finally, there is a concept of interchangeability. Now, this is an even stricter concept than being a biosimilar. Interchangeability refers to a permission to a pharmacist, this is in the US, to substitute a biologic drug for another without consulting the prescribing physician. Now, even a biosimilar is not allowed this in the US unless they have specified and passed even more stringent controls and comparisons with the innovator. So interchangeable biosimilar is expected to produce the same clinical results. So now the onus is clinical, not just biochemical or immunological or laboratory. And there are tougher requirements by the US FDA. Now, this issue, unfortunately, has not been addressed at all by the CDSCO. And most physicians, in fact, also, and pharmacists assume that if a biosimilar comes, it is freely interchangeable with another biosimilar or the innovator, which is absolutely not the case. There are nomenclature issues also which complicate the matter. Most countries with a sensible drug naming policy name a biosimilar in a very different way. In the USA, they add a 4-letter suffix to the brand name, adalimumab XXXX. Or in Japan, they add the suffix BS, biosimilar, to the name so that the person who's buying knows clearly that this is a biosimilar or a copy. However, there is absolutely no change in name in India. So, I mean, it's no surprise that physicians and patients and pharmacists think that it's the same thing and they can substitute it, which is not the case. Labeling issues are there. They don't have adequately detailed information, and whether the data generated was on the innovator. Many biosimilars just copy the data generation part of the product insert from the innovator and just paste it into their product, and they're allowed to market it, sadly. And also the extent of similarity is also not documented. So these are all various issues with biosimilars which really put a doubt in a clinician's and a pharmacologist's mind whether they are actually the same as the innovator, and whether they'll consequently be as safe and effective as the innovator. So these Indian drug policies, although they've made these biologics available and affordable to a large proportion of our population, which is a good thing, but our rules are less stringent. And we are taking a chance in terms of them being non-similar. And they are a heterogeneous bunch of products. Some of them are held to a higher standard. Some of them have passed those standards, but they look absolutely the same. They have the same brand name. There's no way for a lay person to know which ones those are. And others are absolutely which have passed through, especially the ones approved earlier, have passed through much less stringent rules. And since nomenclature is no different from the originator, we don't know. So there's an urgent need for establishing registries for all these intended biosimilars so that we can generate some data and see whether the efficacy and the side effect profiles are similar to the originator or not. And pharmacovigilance needs to be tightened up a lot more so that we can pick up right now these possibilities, since these drugs are not very, very widely prescribed for cost reasons. We might be missing, these adverse effects might be falling through the gaps. We might not come to know unless something, drastic or bad happens. So this is basically what needs to be done, that we need to tighten our regulations. We need to tighten pharmacovigilance to make sure that our patients get the best possible quality of similar biologics that we can give them. Thank you very much.
Thank you, Dr. Abir. Wonderful, wonderful lecture, and the entire gamut of challenges with Indian biosimilars and similar biologics have been covered by you. Really appreciate it. India, we are supposed to be a land of generics, actually. And then you have the branded generics. I'm talking about the new chemical entities, and then you have the branded biosimilars now. So it's unique and different. So there are two questions for you. I think one of them you've actually already answered, but why do you think that the Indian pharmaceutical industry has not opted for the more stringent pathways?
I mean, it's quite obvious. If the government and the CDSCO doesn't require you to jump through various hoops, why would you? I mean, they would take the easiest route of least resistance because it's costly to do these. It's time consuming, and if our rules have inbuilt loopholes or laxities or discretionary powers given to our regulators where they can, waive off certain rules in certain cases, then those will be used. I guess that's my best guess. Why would they work harder than they have to get a regulatory approval?
The point about the cost of development that you made just now is possibly equally important, because on the one hand, you did mention access, they provide access, but the cost of developing a biosimilar is possibly much, much higher than a new chemical entity, and therefore the investments that go in and so forth. The second question is a little different. Have you seen adverse reactions in your practice with the use of biosimilars, which are completely different from an innovator?
No, I haven't. Completely different, no. But we all have seen, all clinicians have seen. I'll give you some examples. There are an inadequate efficacy is a problem. Inadequate efficacy we've definitely seen, especially with rituximab biosimilars in patients with pemphigus, for example, where the biosimilar doesn't work, and when we give the originator, it works spectacularly well. And the other problem, which is a bigger problem, is immunogenicity or secondary failures. So we have so many biologics available with us, which probably work for the first few weeks, but then as you give them, the efficacy drops off very dramatically. So these are all the signs of basically anti-drug antibodies or basically a secondary failure being developed. So these are immunogenic drugs, proteins, and probably not sufficiently similar to the originator and more immunogenic. And this is very common. This we all have seen, that intended biosimilars, some of them tend to have much higher failure rates, especially secondary failure rates. They work well initially, but then they lose efficacy.
Great. Thank you, Dr. Abir. I think that was the last question. Sincere thanks from the department and from the Pharmacovigilance Program of India. Wonderful, wonderful talk, and I look forward to fruitful interactions in the future as well. Thank you so much, Dr. Abir.
Thank you very much. It was a pleasure.
Dr. Samir gives you his regards.
Yeah, please convey my regards to him as well. Thank you very much. Bye-bye.
Bye. And I'll now request Vijaya to propose a vote of thanks and close the meeting.
Yes. Thank you very much, sir and ma'am. With this, we come to the end of our workshop. We hope that the talks have been fruitful and will take your understanding of pharmacovigilance ahead. We had almost 250 plus delegates attending the sessions. We thank all the delegates of the workshop, without whom this would have not been a success. Also, I would like to thank all the faculty members and resource persons who took time from their busy schedule to train and equip all the participants with knowledge and skills on various emerging arenas of pharmacovigilance spectrum. We would like to thank our Dean, Dr. Sangeeta Rawat Madam, and our Academic Dean, Dr. Harish Pathak Sir, for giving us the opportunity and providing us the support to conduct this workshop. We also want to extend our gratitude to heads of department, Dr. Narkal, HOD, Department of Medicine, Dr. Karande, HOD, Department of Pediatrics, Dr. Chandrakala, HOD, Department of Hematology, for their continuing help and guidance toward pharmacovigilance program at our Center. On all our behalf, I would like to extend our sincere gratitude to Dr. Geeta Natraj Madam, Emeritus Professor, Department of Microbiology, Mr. Tushar and Mr. Badal at their Regional Research Center, National Knowledge Network, Government of India, which is situated at KEM, for their constant support throughout the workshop. Dr. Nilima Shirsagar Madam was also supposed to be the part of the workshop, but due to her travel commitments, Madam unable to join the workshop. But she has sent her best regards to everyone. We hope this endeavor will hold its stead and we continue to meet you for such learning opportunities. With this, on behalf of Department of Clinical Pharmacology, I, Dr. Vichaya, declare that the workshop is closed and wish you all happy and pleasant weekend. Thank you.